कांबळे सर,
हा
तुमचा माईक म्यूट करा, तिथे खरखर वाजते काय.
माझ्यात नाही काय वाटत.
नाही ना.
नाही
नाही ते हे होते अजित देशमुख खाली कोण जॉईन झाले त्यांचं वाजत.
येस ओके सर. हॅलो जगताप सर.
हा सर.
हा आपण 7:30 सुरू करूया.
नाही अजून वाघमारे सरांना 10 मिनिटं लागतात पोचायला. मॅडमना पण फोन करतो ना.
होय का. बर
मॅडम अजून जॉईन व्हायला पाहिजेत ना, अजून पार्टिसिपंट कमी आहेत ना.
बर होतील ना.
हो एक 8 लोक तर आहे ना आपण.
हो ठीक.
त्यातली आपली 4 जण आहे आपण.
1 2 3 4. हो
हा चालेल.
चालेल ठीक.
मॅडमना फोन करतो मी.
हो चालेल. खाली हिरवे ना खाली सॉस.
परीक्षा काहीच दिवस होती. समोर म्हणजे काय झालं? ओळख कुठे झाली तुमची? ओळख कॉलेजमधली सुटी.
तो उपडे मॅडम हॅलो
थोडी म्यूट करा.
बोबडे मॅडम आपला माईक म्यूट करा प्लीज. सुधीर कांबळे
हा येस सर.
माईक म्यूट करा.
हा
चालू करूया का सर?
अजून मॅडम जॉईन नाही.
चेअरमन फोन झाला नाही.
हा मॅडम जॉईन हो 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 चालेल. चालेल हो 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 गुड डे. 10 मिनिट.
गुड मॉर्निंग सुहास.
गुड मॉर्निंग. मॉर्निंग सर.
2-3 काढल्या प्रिंट सगळं घेऊन.
हा बघू. हा एक 10 मिनिटं. हा राठोड मॅडम जॉईन होते ते 10 मिनिट. तुझा आवाज येणार व्यवस्थित. हा रेंज मध्ये.
खान
त्यासाठी मी काय करतो.
आता पण काढले 4-5 सगळ्याच फाईल मध्ये घालून.
काय करते आता काय कर.
जरा व्यवस्थित काय 5 मिनिटं नाही नाही. या.
गुड मॉर्निंग. गुड मॉर्निंग. जगताप सर चालू करायचं.
सुहास सर,
हा बोला.
मॅडमला एक 2-4-5 मिनिटं लागतात.
ठीक आहे, ठीक आहे.
एक 5 मिनिट वेट करूया लगेच सर.
ओके ओके.
सोरटे मॅडम आपला माईक म्यूट करा. सर्व पार्टिसिपंटना विनंती आहे की आपापले माईक म्यूट करा.
डायरेक्ट म्यूट ऑल करा.
प्रोग्राम कुठे
सेटिंग मध्ये असेल.
रेकॉर्ड येतात.
सिक्युरिटी मध्ये सिक्युरिटी मध्ये जाऊ. सिक्युरिटी
डॉक्टर अनिल तुमच्याकडच्याच गप्पा ऐकू येतोय.
हा बोला की सर.
गुड मॉर्निंग जय सर.
गुड मॉर्निंग, गुड मॉर्निंग.
हा
पार्टिसिपंट्स मध्ये जाऊ.
भागवत देशमुख सर, गुड मॉर्निंग.
गुड मॉर्निंग सर.
काय मॅडम, एक 10 मिनिटं लागतील मॅडम गाडी आणि मग मला सर पण पोचायचं आहे. चालू करतो हे. आवाज नाही येत. आवाज म्हटलं.
हा सर तुमचा आवाज येतोय.
हा आला 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 हा.
बालेराव सर
हा हा हा
येतोय तुमचा आवाज.
येतोय ना. ओके ओके.
येतोय येतोय येतोय. एक 5-10 मिनिटात सर जॉईन आपण चालू करूया.
हा येस येस येस.
हा राठोड मॅडम जॉईन होत आहेत विद्यापीठातून.
एक कोण कोण जॉईन होते म्हटले. हा
चीफ
आहे
गेस्ट
सर, आहे. मी घेतो.
ओके ओके
झालं.
सुहास सर
हा सर बोला.
मॅडम जॉईन झालेत वाटतं.
चालू करू द्या.
हा एक 2च मिनिटं.
पुढे. हा सर जॉईन होतो. मग आम्हाला सांगायचं नाही तुमच्यासाठी थांबलो. हो 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 हो. सर करून घेऊ चालेल. ओके. चालू करायचं सर.
सुहास सर
हा येस सर.
चालू करा. वाघमारे सर देशमुख सरांचा मोबाईल तिथूनच बोलणार आहे.
येस सर. मॅडम जरा
चालेल तर चालू करा.
मॉर्निंग टू एव्हरीवन. Myself Dr. Suhas Kamble on behalf of Department of Botany, Dahiwadi College, Dahiwadi. I cordially welcome all the delegates, BOS chairman, BOS members, resource persons, chairpersons, and teacher participants to this one-day online workshop on revised syllabus CBCS of B.Sc.III in Botany Practical 3rd and 4th. This workshop includes 4 segments. These are inaugural function. There are 2 sessions and a valedictory function. Each session there will be 2 resource persons and chairpersons, and finally there will be a valedictory function.
Respected and dear all, let us begin with the inaugural function. For this purpose, I would like to request our head of department, Dr. B.V. Kamble sir, to introduce and welcome the respected delegates of this inaugural function. Please, over to
Thank
Dr.
you.
B.V. Kamble sir.
Thank you, Suhas, sir. Am I audible, sir?
Yes, sir.
Today, on the occasion of online workshop on revised syllabus of B.Sc. Part 3, Practical 3rd and 4th, I cordially welcome on behalf of Department of Botany, Dahibadi College, Dahibadi. It gives me immense pleasure to organize such academic workshop, especially for practicals. We will be more happy if university allows us to organize this workshop in offline mode. But anyways, it's okay. Once again, I warmly welcome you all, our
एक मिनिटासाठी येऊन जा.
all resource persons, chairpersons of respective sessions, Professor Dr. B.D. Jadhav, Head of Department of Botany, Shivaji University, Kolhapur, Dr. M.B. Wagmare sir, Chairman, Board of Study, Botany, and my colleagues in the department. Now, it is my prime duty to introduce today's guest for inaugural function. I will introduce Professor Dr. B.D. Jadhav ma'am, Dr. M.B. Wagmare sir, and Principal Dr. B.S. Balwan sir in brief. Professor Dr. B.D. Jadhav ma'am is the chief guest for today's inaugural function. Presently, she is head at Department of Botany, Shivaji Kolhapur. She has academic qualification, MSc and PhD, and he received both degrees from Sant Gadge Baba University, Amravati. She has teaching experience of 22 years. Her research experience is more than 15 years. 8 students have
आवाज कट होतो.
Sir, your voice is not audible.
Yes, sir. Yes, sir. There are some network problems. Okay,
It's clear.
Am I audible, sir?
Yeah, it's clear.
Yes.
Yes, sir. Thank you, sir. Professor Dr. B.D. Jadhav has research experience of more than 15 years. 8 students have received PhD degree and 5 students are doing PhD under her able guidance. She has also published more than 40 research papers in various reputed national and international journals. He is a chairman of B.Sc., Botany, and also working as subject experts and referee for PhD. Dr. M.B. Wagmare, today's keynote address, presently is the chairman of Board of Study Botany, Shivaji University, Kolhapur. He is a member of Academic Council, Shivaji University, Kolhapur. He has 54 publications in various reputed international and national journals. 3 students have received PhD degree and 5 students are presently doing PhD under his guidance. Workshops, seminars attended by him in India and abroad are more than 85. He has attended one conference abroad, that is at Bangkok, and he is also an author for 4 books, and he has organized 13 different conferences. And today's our principal is in charge principal Dr. B.S. Balwan sir. He is the president for this inaugural function. He is MA PhD in Hindi. Presently is in charge of our college, in charge principal, Dahibadi College, Dahibadi. He has 28 years of teaching experience, and he has more than 10 research publications in national and international reputed journals. And he has organized more than 10 conferences and workshops at national and international level in our college. Along with this, I welcome all the principals, all board of study members, professors, and heads of departments of botany and teachers, friends. Along with this, I welcome today's resource persons, Dr. Sagar Deshmukh, Dr. Sanjay Jadhav, Dr. Sohan Moholkar, Dr. Babu Sonar, and the
बैठ सकती हूँ क्या यहाँ
chairperson
पे?
for respective sessions, Dr. E.B. Bhalerao, in charge principal, Kisabia Mahavidyalaya Y, Mr. Samson Gaikwad, Dr. S.K. Khare, and Dr. Rekha Devkar, and Principal Dr. S.D. Helkar from Professor N.D. College, N.D. Patil Mahavidyalaya, Malkapur. I welcome you all once again, and my request Professor Dr. B.D. Jadhav ma'am to deliver her speech on this occasion. Thank you. Over to B.D. Jadhav ma'am.
Dr. Suhas, you can invite her again.
Yes, sir.
You can invite Dr. Jadhav madam again. Probably he might not be listening.
Yes. Yes. I request Professor Dr. V.D. Jadhav ma'am to wish us on this practical.
हॅलो कांबळे सर. कांबळे सर.
येस सर.
काय अडचण येते का?
नेटवर्क इश्यू आहे सर.
हॅलो.
हॅलो सर.
नेटवर्क इश्यू आहे सर, 1-2 मिनिट.
सुहास सर.
येस सर.
हा मॅडम जॉईन झालेत बघा आता त्यांना.
येस येस.
तो आज परत अनाउन्समेंट करा, मॅडमला इन्व्हाइट करा.
येस येस.
बोला बघ.
I request Professor Dr. V.D. Jadhav Madam to guide us on this practical workshop.
मॅडमला बहुतेक आवाज जात नसतो आपल्या इकडे असं वाटतं.
जय सर.
प्रॉब्लेम आहे.
मॅडमना आपला आवाज जाईना झालाय.
आपला आवाज जाईन असं दिसतंय. मॅडमला कॉल करा डायरेक्ट.
केला 2 वेळा पण मॅडम पुन्हा जॉईन झाल्या तरी देखील जाईना झाला. मी कॉलच करतोय.
जगताप सर.
हा आवाज येईन. आपल्याला इन्व्हाइट केले कॉल आपल्या टॉकसाठी.
हॅलो कांबळे सर.
हा बोला की.
हा आवाज येते का माझं बघा कांबळे सर. हा ओके.
जाधव मॅडम बाहेर पडून रिकनेक्ट होत आहेत. त्यांना आवाजाचा काहीतरी इश्यू आहे, ऑडिओ प्रॉब्लेम आहे. कनेक्ट होत आहेत. मॅडमचा कॉल झाला आता.
अनम्यूट केलंय मॅडम ना.
हॅलो कांबळे सर.
हा सर.
हा मॅडमचा मेसेज घ्या मागून आणि ते रीड करा नंतर ना, काय अडचण येणार नाही. हॅलो.
हो ठीक आहे सर.
हा बघा होते काय जॉईन.
ओके.
हा
ओके 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 ठीक आहे.
Hello.
Yes ma'am.
I am audible to you?
Yes ma'am, you are audible.
Okay. So can I start my speech?
Yes.
Okay. So today's distinguished guest, Dr. B.S.Balwan, Principal of Daivadi College, Dr. V.V. Kamble, Head Department of Botany, Vice Chairman, Dr. M.B. Wagmare, Coordinator, Dr. Anil Jagtap, and colleagues from the various colleges. So, first of all, I am very much thankful for giving me an opportunity to address as a chief guest for today's online workshop on revised syllabus of B.Sc.III Botany. It gives me immense pleasure to convey you all that Dahibadi College, Dahibadi, District Satara, is hosting the online workshop on the revised syllabus of B.Sc.III Botany on practical number 3 and practical 4. I am pleased to see that the Department of Botany has brought together all the teachers of botany from various colleges for said workshop. This workshop will address the need for exchange of information regarding the revised syllabus among the experts on practical third based on paper number 11 and paper number 16. Paper number 11 this is cytology and research techniques in biology, and paper 16 biofertilizers and herbal drug technology. Similarly, practical 4 based on theory paper 12 and 13. 12 paper is horticultural and gardening, and 13 paper is a plant biochemistry and molecular biology. So first objective of this workshop is to develop the skill of regarding the practical in the students about how to handle the experiment, laboratory material and equipments, along with to provide the practical experience to the students as a part of the course to develop scientific ability to work in the field of research and other fields of their own interest and to make them fit for the society. So regarding this, the workshop, today's workshop, exchange the view among the expert on practical third and fourth. So I congratulate the principal college, Dahibadi College, Dahibadi, and his entire team for organizing an online workshop on B.Sc.III Botany syllabus. On behalf of my department and Shivaji University, I extend a warm welcome and grand success for today's workshop. Thank you. Thank you very much. And all the best for this workshop.
Thank you very much, madam, for your good wishes. I would like to request Dr. M.B. Wagmare sir to deliver keynote address.
Distinguished participants. Hello, आवाज ऐकू येतो माझा? Hello, can I audible?
Yes,
Hello.
sir.
Okay. Yes, sir. Yes, sir. Distinguished participants, ladies and gentlemen, good morning to all of you. With prior permission to the Shivaji University Kolhapur, Dahibadi College, Dahibadi organized a one-day workshop on the revised syllabus of the B.Sc.III Botany, practical number 3 and 4. For this moment, the chairperson of the inaugural function, Dr. Balwant sir, principal of Dahibadi College, Dahibadi, chief guest for this function, Professor Dr. V.D. Jadhav Madam, Head Department of the Botany, Shivaji University, Kolhapur. Coordinator of this workshop, Dr. V.V. Kamble, Organizing Secretary, Dr. A. Jagtap sir, Resource Person, Dr. S.Y. Jadhav sir, Dr. S.M. Moholkar, Dr. S.A. Deshmukh, and Dr. B.A. Sonar, and all dear participants. It's a great honor to me to deliver the keynote speech at this very important forum. First, I would like to congratulate the principal and all the organizing committee of this workshop on the behalf of the Shivaji University Kolhapur for the excellent arrangement of workshop. In the last week, KWC College Sangli organized the workshop on the practical number 1 and 2. So today we are going to discuss on the practical number 3 and 4. In the practical number 3, there are 21 experiments, and same number of the experiments are present in practical number 4. To the resource person, it is very difficult to explain all the experiment in an hour on the online mode. So I am requesting to the resource person to focus the experiment which are newly introduced into the syllabus. We have made the discussion on the newly introduced experiments and practicals. For example, one practical is introduced in practical number 3, that is Beers and Lambert's law. In this experiment, what we accept from the resource person, that is only you have to try to say the principle of this experiment, which material we can take for this experiment, and what is the main significance of this experiment. Because I think according to my knowledge, this experiment is first time introduced in our syllabus. Actually, this experiment is the photochemistry practical, but why we have introduced this practical in the TY B.Sc. Botany syllabus? Just we think and try to focus on these above point just I have mentioned to you. If you try to give the concrete information regarding this, then we'll get the information, and that will be very easy to conduct the practicals in our laboratory. Another one important practical is introduced, that is a very simple title you have mentioned, that is a cytological slide. So on this, what we need to discuss, that is which plant material we take for the preparation of the cytological slides. Otherwise, in my experiment, what I observed, once the students prepare the slide and they keep the slides as it is till 5 years. So I request to all the teachers, we don't do such things. Every year you have to prepare the new slides and give the chance to prepare the slides to the students. Similarly, in the practical number 4, all the experiments are very student centric. So here I would like to suggest and request you all, try to conduct the experiment in the field area, because just I gone through the syllabus into tomorrow morning, in the today morning, then I find that in the horticulture, there are the number of the practicals are based on the gardening. So why not we carry the practicals in the field instead of carrying the practicals into the laboratory? So once again, I request to all the participants and resource person, focus only on the newly introduced practicals and give the information in detail regarding to carry the practicals in a very easy manner. Once again, I congratulate the organizing committee and thank you very much for your kind attention. Wish you all the best. Thank you. Thank you, sir.
Thank you so much, sir. Now I would like to request Principal Dr. B.S. Balwan sir to guide us with their presidential speech.
हा
साहेबांना हे करायचं ना.
उघड
कोणाकडे तरी देऊन हेच करायला पाहिजे. हॅलो सर. हॅलो. हॅलो.
आवाज येतोय सर? येतोय आवाज.
माझा आवाज येतोय का सर तुमच्यापर्यंत?
हो सर हो.
दहिवडी कॉलेजमध्ये आज शिवाजी विद्यापीठ कोल्हापूर आणि वनस्पतीशास्त्र विभाग यांच्या संयुक्त विद्यमाने B.Sc. भाग 3 प्रॅक्टिकल या विषयावर आयोजित केलेल्या या कार्यशाळेच्या प्रमुख पाहुण्या माननीय डॉक्टर P.D. जाधव मॅडम, या कार्यशाळेचे बीजभाषक आणि माझे सहकारी मित्र माननीय डॉक्टर S.B. वाघमारे सर, या विभागाचे विभाग प्रमुख प्राध्यापक डॉक्टर कांबळे बीबी, त्यांचे सहकारी डॉक्टर जगताप सर, डॉक्टर सुहास कांबळे, प्राध्यापिका नाळे मॅडम आणि या कार्यशाळेसाठी ऑनलाईन सहभागी झालेले शिवाजी विद्यापीठाच्या कार्यक्षेत्रातील सर्व सहकारी प्राध्यापक आणि भगिनींनो, मी आपल्या सर्वांचं महाविद्यालयाच्या वतीनं आणि वनस्पतीशास्त्र विभागाच्या वतीनं मनपूर्वक स्वागत करतो. आणि सुरुवातीलाच शिवाजी विद्यापीठाला धन्यवाद देतो की ही कार्यशाळा आम्हाला आयोजित करण्याची संधी दिली. त्याचबरोबर शिवाजी विद्यापीठाच्या वनस्पतीशास्त्र विभागाचे चेअरमन माननीय प्राध्यापक डॉक्टर S.B. वाघमारे आणि त्यांचे सर्व सदस्य यांना देखील धन्यवाद देतो की त्यांनी B.Sc. भाग 3 प्रॅक्टिकलची ही कार्यशाळा आयोजित करण्यासाठी आम्हाला जी संधी दिली आहे, या संधीचा निश्चितपणे आमच्या महाविद्यालयातील विद्यार्थ्यांच्या बरोबरच सातारा, सांगली, कोल्हापूर जिल्ह्यातील वनस्पतीशास्त्र या विषयाचा अभ्यास करणारे जे विद्यार्थी आहेत, या विद्यार्थ्यांच्या पर्यंत निश्चितपणे आज या कार्यशाळेच्या निमित्तानं दिली जाणारी जी माहिती आहे, ही माहिती पोहोचेल याबद्दल माझ्या मनामध्ये कोणत्याही प्रकारची शंका नाही. या कार्यशाळेच्या निमित्तानं आज दिवसभर जे मार्गदर्शन करणारे मान्यवर आहेत, त्यामध्ये माननीय S.A. देशमुख सर, त्याचबरोबर दुसरे रिसोर्स पर्सन माननीय श्यामसन गायकवाड सर, त्याचबरोबर माननीय डॉक्टर S.M. मोहळकर सर, या सर्वांचं मी महाविद्यालयाच्या वतीनं मनपूर्वक स्वागत करतो. त्यांना धन्यवाद देतो. या कार्यशाळेसाठी आपण आपला मूल्यवान वेळ आम्हाला दिलेला आहे आणि याचा निश्चितपणे शिवाजी विद्यापीठातील सर्व प्राध्यापक बंधू भगिनींना याचा फायदा होईल. आजच्या कार्यक्रमाचे प्रमुख पाहुणे माननीय डॉक्टर P.D. जाधव मॅडम, माननीय डॉक्टर M.B. वाघमारे सर आणि सर्व विषय तज्ञांचं पुन्हा एकदा महाविद्यालयाच्या वतीनं मी मनपूर्वक स्वागत करतो आणि त्यांना धन्यवाद देतो. आणि या ठिकाणी मी थांबतो. या कार्यशाळेसाठी महाविद्यालयाच्या वतीनं आपणा सर्वांना मनपूर्वक शुभेच्छा. धन्यवाद सर. हॅलो.
ओके, थँक्यू सर.
Dr. Jagtap sir, please propose a vote of thanks.
Okay, good morning to all of you. So firstly, I thankful to University Authority for giving the chance to organize the workshop of revised syllabus of B.Sc.III. Practical number 3rd and 4th. So I propose a vote of thanks towards a Professor Dr. V.D. Jadhav ma'am to join with us and to give a valuable guidance in his busy schedule. So I will also thankful to Dr. M.B. Wagmare sir, BO Chairman in Botany, Shivaji University, Kolhapur, as well as our visionary and dynamic principal, Dr. B.S. Balwant sir, and all the delegates joined to this workshop. With these few words, with the kind permission of President, now I declare the inauguration function of this program is over. So thank you very much. Sir, please.
Thank you, sir. Thank you very much. We are going to start session first. Now, I would like to resource person of this session, Dr. S.A. Deshmukh sir, who is now working at the Department of Botany, the New College, Kolhapur. He has completed MSc as well as MBA degree. He has completed PhD degree under the guidance of Honorable Dr. D.K. Gaikwad sir. In spite of this, he has qualified SET, NET and GATE exams. He has 15 years of teaching experience. He has published 22 research papers in national and international research journals. He has completed 2 minor research projects. We have Chairperson Dr. E.B. Bhalerao, Sir, in charge principal, Kisanveer Mahavidyalay Vai, as a chairperson for this session. With the kind permission of chairperson, I request Dr. S.A. Deshmukh sir to present his presentation based on practical 3. Please, sir.
Okay, sir. You can proceed further.
Deshmukh sir.
Thank you, sir. Is it audible to you?
Yes, sir.
Yes, sir. You are audible.
Okay, thank you. So, first of all, I am very much thankful to all the faculty member as well as principal, Dahivadi College, Dahivadi, for giving me an opportunity as a resource person. As per my personal opinion, apart from the nature, no one is the resource, but you have given me the chance to be a resource. So once again, I am very much thankful to you. And here we will discuss about the practicals which have been newly introduced in our syllabus for B.Sc.III. So I am here presenting the screen, and is it visible? Screen is visible? Yes or no?
Yes, sir. Visible.
Okay, thank you.
But
So
please take it.
here
Okay, sir.
during this online workshop on revised syllabus as per CBCS, we have to study various practicals which have been there in practical number 3. All these practicals, they have been based on paper number 11 and paper number 16. So the organizing committee, they have given me the practicals from 1 to 12. I request all the participants to unmute yourself.
मोहळकर सर आणि आवळे सर तुमचा माईक म्यूट करा.
Sorry, I request all the participants to mute themselves. So here we have to study in practical 3. We have to study the practicals allotted to me. They are from practical number 1 to practical number 12, and I will just read all those practicals. Practical 1, it relates with the prokaryotic and eukaryotic cell. Practical 2 relates with the photomicrography technique, and practical number 12, it also relates with these practicals, that is submission of photomicrography. Practical 4 relates with the cell structure of onion, hydrilla, and spirogyra. 5th and 6th, they relate with the cytological techniques, mitosis and meiosis cell divisions. Number 8, micrometry technique. Number 9, DNA packaging by micrographs. 10 Beer and Lambert's law, 11 preparation of permanent cytological slides. If you overlook all these numbers or all these practicals, you will come to know that all these practicals, apart from practical number 10, that is study of Beer and Lambert's law, all the practicals they have been earlier studied, earlier taught by us. But these practicals might be new to the new teachers. But these are the basic practicals. They relate with the cell structure, eukaryotic, prokaryotic cell, as well as mitosis, meiosis, and DNA packaging and permanent slides. One addition is there, that is nothing but the Beer and Lambert's law. So we will, as per the guidelines given by the BOH chairman, we will mainly concentrate on Beer and Lambert's law. That is the practical number 10. So we will begin with the practical number 1. Name of this practical is study of prokaryotic and eukaryotic cells with electron micrograph and photograph. In the present scenario, if you think about this practical, don't take it lightly, but this practical already we have studied, or all the students they have studied at their primary level. So here we have to just give the micrograph or the photograph. Now you are observing two images here. One is the micrograph and other is the image. This is not photograph. Photograph is nothing but the picture taken by an instrument. Here this is the schematic representation of second diagram, which is represented by green color, bacterium with flagellum. That is nothing, not a photograph, but this is an image. For our convenience, if we do not stick up with the title of this practical, we can take this image also, this colored image also. Okay, this is not photograph, that's why can be omitted from the at the time of practical, but for our convenience we can take this image also to study the students related with the prokaryotic cell. The final aim is nothing but to stick up with the micrograph or photograph. The final aim would be students should have to get perfect knowledge or all the knowledge related with the prokaryotic cell. So here we are observing the bacterial cell. We can consider E. coli as an example of prokaryote. Bacterial cell, one might give micrograph or photograph, and in prokaryotic cell, all the structures are there. I request all the participants to mute themselves. Okay, so here we are observing the prokaryotic cell. All the concepts we know about the prokaryotic cells, and here we have to mention the cell wall, capsule, plasma membrane, nucleoid, cytoplasm, ribosomes, flagellum, pillars with respect to bacterial cell, and we have to point out that many cell organelles they are absent, and many cell organelles are absent in prokaryotic cell, and the nucleus is not well defined in prokaryotic cell. That we all of us know. Okay, so the structures in bacterial cell, we have to concentrate on essential structures and particular structures. Essential structures, there are cell wall, cell membrane, cytoplasm, nuclear material, and in particular structures, cell envelope, capsule, flagella, and pila. At the time of practical examination, what will be the scenario of this practical? This practically probably will be for identification and comment or describe. So this will maximum it will carry 2 marks, or the marks will be marks given as per the paper setters. So if the practical is for 2 marks, identification will carry 1 mark, and comments and description will carry 1 mark. At the time of practical as well as at the time of regular practicals, at the time of practical examination as well as regular practicals, we have to provide or we have to give the photograph of this bacterial cell, and we have to ask the students to recognize all the structures or all the structural components which are present in the prokaryotic cell. In next slide, I have mentioned all the details related to the prokaryotic cells that I will not read out. Okay, those details they have been given for our convenience. Here, I will not read all the details. The details are related with the essential structures of prokaryotic cell and particular structures. These are the details which might be read by yourself later on. Okay, then in the same manner, another practical is there. Before that, if one has the question, now if someone has the question while teaching the plant science, why not to study the plant as an example of prokaryotic cell? Then there is also a solution. You might take the example of blue-green algae, nostoc. But in current scenario or at present literature, they are also nostoc is also considered or grouped in cyanobacteria. They are the plant aliens. Uh, One can consider nostoc as plant aliens. It shows both the characteristics of bacteria as well as algae. So in recent literature, they have been also grouped in cyanobacteria. Next, in the similar manner, second practical is study of eukaryotic cells with electron micrograph or photograph. Here we have taken plant cell for a as an example. So among these three, one can use any one of the image, and it will be better if we use or if you give color photographs at the time of regular practicals as well as practical examination. In this plant cell or the detailed account of plant cell, either in micrograph or photograph. Already I have discussed the meaning of photograph as well as an image. So for our convenience, we can give these images also to the students, and these images they will be better to the students to recognize as well as to study all the cellular details present within a plant cell. Here, if you observe the image, any one of the image right below, uh, uh, in that image, the crystals are they have also been mentioned along with the cell organelles, chloroplast, mitochondria, Golgi complex, endoplasmic reticulum, amyloplast, starch grains, and other crystals, rapid crystals. They have been also mentioned. So the students will come to know not only cell organelles but certain other secretory or other related compounds or structures. They are also present within the cells. But at that time, we have to give the example with related to ficus leaf or uh, colocasia leaf with respect to rapid crystals. Okay, same this practical will also it will be also for about identification and comments and describe. It will carry certain amount of marks, either 2 or 4 as per the paper setters, and the marks will be distributed as identification 1 mark if the practical is for about 2 marks, and comments and description for 1 mark. At the time of exam, we can point out any one of the cell organelle or the cell structure, and the students will write about that particular cell structure or cell organelle. Okay, so these are the plant cell structures. I will not again read out all the structures, but I have put all the structures. That's why it will become easy to everyone when I will share those slides to the organizing members. Okay, in plant cell structure, cell wall, cell membrane, nucleus, chloroplast, central vacuole, Golgi apparatus, mitochondria, lysosomes. All these are most all these are very basic information related with the plant cell structure that all of us know. That's why I will not go into detail of this plant cell structure. Okay, so next practical, again it relates with the cell structure. This practical is study of cell structure in onion, hydrilla leaf, and Spirogyra filament. To complete this practical, we have to we must have all those equipments as well as instruments. Okay, so glycerin, safranin solution, dropper, forceps, and other materials there should be present at the time of this practical. So first we will discuss about study of cell structure in onion. Here this diagram represents detailed account how to perform the practical of cell structure in onion. We have to cut the onion into equal quarters. If they are not equal, that doesn't matter. One of the fleshy scale leaves is removed from the onion, and that fleshy scale leaf is snapped back to expose the epidermis. Then a thin layer of this epidermis is peeled off, and that thin layer of epidermis is stained with dilute safranin, and after proper staining it is washed with water and mounted in a drop of glycerin. Now we will observe the cell structure like this, the columnar or elongated cells of the onion, we can observe the cells are regularly shaped, and they lie side by side. And each cell has a distinct cell wall, and we can observe a prominent nucleus within that particular onion cell. So precautions should have to be taken during performing this practical, and these precautions relates with the material should not have to be dry. Second one, staining of peel should neither be too dark nor too light. An extra stain that have to be removed by the blotting paper. Then and then only we will observe the clear look of that particular onion cell. Otherwise, what will happen? All the procedure we will perform in perfect manner, but the staining is not proper, as well as that plant material, if it get dry during the course of practical, we will not get better results, or we will not observe the onion cells in their original shape also. Okay, so second practical, it also relates with the cell study. Here the plant material has been changed, and the plant material is hydrilla leaf cell. We have to observe the cell structure of hydrilla plant. I will give one hint related to the hydrilla plant. If you collect the hydrilla plant in water bodies. Nowadays, due to overpollution of water bodies, over the lip surface of hydrilla, you will find another microorganisms or other algae. So at the time of practical, what will happen? We will observe another structures or other structures apart from the cellular details of the hydrilla cell. For that purpose, we have to concentrate. Just take the leaf from the upper rosette of the overlap unopened leaves. You are observing the hydrilla twigs here, and at the apical region or the apex region, the leaves are in rosette form. So just we have to expose the leaves, and we have to take the leaves which is slightly lighter than the other leaves, because it is in contact form, it is unopened, and this leaf, it is not polluted or it is not covered with other microorganisms rather than the other open or whole leaves. Okay, so here the glass slide is taken. As I mentioned, just select the proper leaf, so that other microorganisms should not get come into over that slide, and we can't easily visible the cell structure of hydrilla. So this is the hint. All the details all of us know related with the hydrilla leaf structure. Now hydrilla leaf structure, the slide will look like this. The slide will be observed like the wall consisting of bricks, and in that particular hydrilla cell we will observe the chloroplasts also. Okay, so next practical, it relates with the cell structure in Spirogyra filament. Just we have to collect the Spirogyra, and with the help of needles, we have to separate the Spirogyra filaments. First, utmost important thing is that the Spirogyra filaments, they have to properly separated from each other, so that a single filament can be easily visible. Otherwise, what will happen, if you put all the Spirogyra on into the slide, we will not observe the cellular details uh, with high efficiency. So we have to separate the filaments before performing the practicals, and we have to, after separation of single filament, we have to observe that single filament under the microscope. At that time, we will observe a single cell structure, and that single cell structure will show all the details. Important or interesting thing related with the students, they are eager to observe, or they are eager to look after the spiral or ribbon shaped chloroplast in Spirogyra. This is the important or interesting thing with respect to students. Uh, okay, so we have to mention, or we have to show the ribbon shaped chloroplast to the students. Again, this practical, it will be either for table work, or practical will be for identification in comments or discussion. Okay, after completion of this practical, another practical that already we have taught at B.Sc.I level, or in earlier syllabus, it was for B.Sc.III also. So study of mitosis. All of us, we know that mitosis is a type of cell division in which single parental cell, that is vesicle cell, divides to form two daughter cells, which are exactly similar with each other and to the parental cell. The aim and objective of mitosis is nothing but the growth and development of particular organism. Here we are going to study the mitosis in onion cells. So we have to buy the onions from the market. Those onion bulbs should have to be healthy. Then, with the help of sharp blades, carefully snip the dry roots of the onion. And after snipping the dry roots of the onion, we have to put the onion over a beaker or a conical flask or a container containing water. So after a week or after 4 to 6 days, what will happen? That onion from the point of snipping, it will form the roots. It will generate the roots, and those roots, from those roots, we have to collect the healthy roots, and those healthy roots, after those healthy roots, we have to cut, and we have to store them in aceto-alcohol solution. That solution consists glacial acetic acid and ethanol in the ratio of 1:3. So allow these root tips to remain in the vial or in the mixture for 24 hours. Then, with the help of forceps, pick one root and set it on a new glass slide. Use 1 drop of N by 10 HCl. Then additionally add 9 to 10 drops of acetocarmine stain and heat the root gently or lightly on the burner. Here precaution should have to be taken. The material should not have to get dry. Otherwise we will not get the proper results. Again, excessive stain should have to be removed with the help of filter paper. The And now the key thing is there. We have to observe the onion root tip, and we have to select the meristematic portion of that onion root tip. And the meristematic portion, if you observe the that particular stained onion root against the light, the darkly stained body, that will be the meristematic root tip. Or just you cut off the meristematic root and discard remaining part of that onion root. Now this is the selection of material for to carry out the mitosis. Again, with the help of needle, a cover slip can be mounted on it. Okay. Here for staining purpose, acetocarmine stain is used. 1 drop of acetocarmine is loaded on the slide, and a cover slip is mounted with the help of needle or forceps. Now we have to squash the material. We have to squash the material. Mitosis is done with the help of squashing technique. Okay, squash technique. With the help of needle, a cover slip can be mounted on it, and gently tap the cover slip with an unsharpened end of a needle in order to separate the meristematic cells evenly over the slide. Now, after the proper squashing, we have to observe the various stages of mitosis under the microscope at 10x magnification, and later we will proceed further for higher magnification purpose. So this is the regular practical that we have already performed during our early course. Okay, so these are the results or observations and conclusions. Now mitosis, here I will skip this slide and we will go to this slide. Okay, so proper selection of the material is very important, that we do, but students sometimes or most of the times they took entire root portion and they squash all the roots and they put the cover slip over that root. They make spindle formation for that root to cover to get inside that cover slip, and they observe the cells that will not work for them. So before starting the practical, one should give this printout, can be given to the students before start of the practical. This will be helpful to them to find out various mitotic stages. Again, very interestingly, when I take this practical to the students, there are certain superstitions among the students. What they feel, I personally ask the students, and they say that, sir, the entire view of the microscope objective is a single cell. They consider the entire view of the microscope objective as a single cell, and they start to find out the chromosome within that entire view. This is one of the biggest superstitions among the students. Okay, this was at B.Sc.III. Then in single view, only one stage is present. They also thought that only one stage, either from prophase, metaphase, anaphase to telophase. Among these four stages, only one stage is visible during that slide. This is the another biggest superstitions among the students. We have to state that, or we have to say that all the stages can be visible in a single slide also. Okay. Now already I have stated, most of the time students took entire route to perform the mitosis. That will not work. Okay. And very interestingly, whenever we ask to the students, apart from the onion root tip, other, suppose mango root is given, it will, does it show mitosis? 80 to 90% students say no. So we have to make student friendly practical, and we have to vanish all the superstitions which are present in the mind of students. So I will once again repeat, these superstitions are not related with the each and every students. This is my personal experience, so they relate with my students also. Okay, so don't take it personally related with the superstitions. But here one implementation can be added, that is, just give this printout before starting the material and label this printout from interface up to the telophase. Uh, we have to guarantee, we have to give guarantee to the students, not warranty, but guarantee to the students. In your slide, at least telophase 1 and prophase, they, sorry, telophase 1, no, in your slide, with respect to mitosis, at least prophase and telophase, they should have to be visible, or you have to observe at least prophase and telophase from the slide, prepared slide. Uh, as per syllabus, we are taking this practical, and again the students, they think that the roots or root apical meristem only possess the, or only have the capacity to divide, that means show, to capacity to show the mitosis. But as an interest, if anyone is interested, here you are observing one image that is of cycas. You are observing young developing cycas. The cycas croziers are there. If you just took the apex of that procedure and you follow whole the procedure, we will observe even we will observe better chromosomes even than the onion also. Okay, I have personally performed this practical. There is no rocket science involved in this practical. You have to just took the material, you have to just fix the material, that cycus tips. Okay, and you have to perform all the practical same in the manner related with the mitosis which we have performed in onions. Also, we will observe large chromosomes, or larger than the onion cell chromosomes in cycus croziers also. So as an interest, as an additional job, we can give this material to the students also, so that they will come to know not only roots but also shoot tip, they divides. This is no doubt, this is universal thing, that RAM and SAM show that mitosis or meristematic, they possess meristematic tissues and they exhibit mitosis. This is universal thing, but students will come to know by means of practical, not only root but also should it show the dividing stages, that is mitosis. Okay. Then next, learning objectives related with the mitosis. Having completed the practical of mitosis, students should be able to define various terms which are related with this practical. So all these terms we are going to teach them in at the time of theory, but at the time of practical, what will happen, students will be asked to perform or to make the slides, and at the time of observing the slides, the examiner might ask certain questions. So for that purpose, we have to look after these all after we have to look after all these questions describe the overall process of mitosis. How will you define the term mitosis? What is the difference in between all these stages? So students should have to know all the terminologies related with the mitosis. Next practical uh, again relates with the cell division. This is study of meiosis. Again, as per the definition, meiosis is a type of cell division in which a diploid parental reproductive parental cell divides to form four daughter cells, which are not exactly similar with each other and to the parental cell. Here, as per the syllabus, we have to study meiosis in onion flowers. We are observing two images. First one is inflorescence of onion. It looks beautiful, but this is not suitable for meiosis because this is not suitable for meiosis practical because already pollen grains they have been developed or formed. The right selection will be you have to choose the inflorescence which is not open, which is entirely covered by the sheath. Okay, so this is the proper selection. If we select the inflorescence uh, in left image and perform all the procedures very carefully related with the meiosis practical, we will not get results. We will observe the pollen grains only. So here selection of plant material is very important. So select appropriate flower buds of different sizes from the inflorescence. Fix them in Caronese solution. This is nothing but the ethanol and glycyl acetic acid in the ratio of 3:1. This is used as fixative. Then take a preserved flower bud and place it on glass slide. Separate the anthers and discard the other parts of the bud. Put 1 to 2 drops of acetocarmine stain. And here, during the process of meiosis, we have to smear the anthers. Then, what is the difference in between squash and smear? Squash technique has been used at the time of mitosis, where we imply certain or apply certain pressure for the uniform separation of the meristematic cells. Here, we have to perform the technique of smear. We have to smear the anthers. Now, the difference in between squashing and smear means we have to puncture the anthers. Okay, we have to puncture the anthers, and so that the uh, meiotic cells or the cells showing division phases related with the meiosis, they will oozes out from that anther and they will come into the medium that is nothing but the acetocarmine stain. Okay, so leave the material in the stain for about 5 minutes. Place a cover slip over them and tap it gently with a needle or pencil. Okay, warm it slightly over the flame. Again, the precaution has to be took, that is, do not dry all the stain over that which is present on that slide. Put a piece of blotting paper on the cover slip and apply uniform pressure with your thumb and observe the slide under the microscope for different meiotic stages. In next slide, I have actually given the procedure. I have actually presented the procedure what we have to perform, that already we have read during in earlier slide with point wise. Okay, just selection of floral bud, then fixation of that particular selected floral buds. In slide number 2, we are observing the separation of flowers. Slide number 3 shows separation of anthers from that particular flower. Slide number 4, a drop of acetocarmine has been put over that slide or that material. Fifth one, with the help of blunt end of the glass rod, the material has been crushed. Or when we use the term smear, with the help of needle, you can puncture the anther lobes also, and cover slip has been put, and we have to observe that particular slide under 10x magnification, and rather we have to proceed further for higher magnification. Then what we will observe? We know that meiosis show two different. Meiosis can be differentiated into two phases: meiosis 1 and meiosis 2. Meiosis 1 show four substages: prophase 1, metaphase 1, anaphase 1, telophase 1. Prophase 1 show five substages. Okay, and meiosis 2 shows four different stages: prophase 2, metaphase 2, anaphase 2, and telophase 2. Now the question arises that all this is very useful at the time of theory. One can remember all those stages from prophase 1 to telophase 2 or from meiosis 1 to meiosis 2 in his or her theory exam. But what will be the scenario at the time of practical? Here student should have to recognize at least one phase, either telophase 1 or telophase 2. Okay, whenever student is going to perform the practical, if metaphase, anaphase and other metaphase 1, anaphase 1, metaphase 2, anaphase 2, if they are visible, that is well and good. But student must have to show, or student must have to observe at least telophase 1 and telophase 2 stages, and along with prophase 1, because this is a very interesting one. How they can easily recognize telophase 1 and telophase 2? Telophase 1, just we have to mention that single cell show two nuclei or two certain dark stained regions at their polar sides, that will be telophase 1. And if the single cell show four compartments showing four prophase like manner cells, that will be telophase 2. Okay, so we have to make convenient the students to observe at least telophase 1 and telophase 2 stages. Okay, once again we can also give this printout to the students so that it will become very easy to the students to find out all the stages, or minimum prophase telophase 1 and telophase 2 stages are. Again, there are superstitions in the mind of students related with the meiosis. They are similar as already I explained in mitosis. Okay, so this is the practical related with the meiosis. Precaution has to be taken whenever we warm the slide. Do not completely dry the slide over the burner or spirit lamp. This is the utmost precaution that we have to take. Okay, so here we complete the process of, or the practical of meiosis. Now the next practical that relates with the micrometry technique. This practical is there this was there in earlier or previous syllabus also. Here you are observing, or we observe scale. Okay, and in regular scale which we have, it show the measurements from 1 centimeter to 15 centimeters, and 1 centimeter show 1 centimeter region, it is divided into 10 micro regions, that is nothing but the 1 millimeter. But did micrometry technique, in micrometry technique, one of the important aspects is that we have to determine the calibration factor. We have to determine the calibration factor. And to determine the calibration factor, this is again not rocket science. This is a simple proportional mathematical equation. There are two stages. One is ocular micrometer, other is the stage micrometer. Okay, we have to remember two stages, two micrometers. One is eyepiece or ocular micrometer, and other is the stage micrometer. Now we have to remember one thing is that the stage micrometer, or the science or the measurements on stage micrometer, they are perfect one, or they have certain dimensions. The stage micrometer, one division of stage micrometer is about 10 micrometer. One division of stage micrometer is nothing but the 10 micrometer. Okay, so known ratio is from, so known, I request all the participants to mute themselves. Okay. So the known ratio is that one division of stage micrometer is 10 micrometer. So this thing is known to us. But from ocular micrometer, we don't know any measurement. But we have to use ocular micrometer for our further study. We have to remove the stage micrometer. So before that, calibration of ocular micrometer or that microscope should have to be done. And calibration is done just desired power objective of microscope was taken into position. Please follow the steps for the calibration of microscope in sequential manner. So remove the eye lens and replace that eye lens with ocular micrometer. Now clip the stage micrometer on the stage. We know one division of stage micrometer is equal to 10 micrometer. This is the known thing with us. Next step is the ocular micrometer or the eye lens. It is rotated to superimpose the marking scales on both the micrometers. Now, just observe this slide, which have been there below the heading of micrometry technique. In this, we are observing eyepiece, that means ocular micrometer, as well as stage micrometer. We have to superimpose the first scale of ocular micrometer with that of stage micrometer, and after superimposing or after coinciding first scale or zero scale of eyepiece with zero scale of stage micrometer, just go on checking the scales or the next measurements which coincide 100% with each other. Here in these scales, here in both the scales, that means eyepiece micrometer as well as stage micrometer, we will observe that 60 number scale coincides, 60 number scale from the eyepiece micrometer exactly coincides with the whatever amount, whatever it will be the number, exactly coincides with this digit of the stage micrometer. Now we have to count the number. This will be 10, 20, 30 and 40. So 40 number scale from stage micrometer, it coincides 100% with the 60 number scale of the eyepiece micrometer. This clearly states that 60 digits or 60 smaller units of ocular micrometer, they corresponds with the 40 smaller units of stage micrometer. Okay, now we know that 1 unit of stage micrometer is nothing but the 10 micrometer. So here we can observe that ocular division 60 is equal to stage division 40. That's why 60 ocular division is equal will be 400 micrometers, since 1 SD is equal to 10 million. This is simple thing or simple correlation ratio. Therefore, 1 OD will be 400 divided by 60, and in this case, the 1 OD will be 6.66 million. This will be the calibration factor for that particular objective or that particular microscope. If you change the objective, again you have to determine the calibration factor for that next magnification factor or magnified objective. Okay, so this will be the calibration factor for that particular microscope. This will be 6.66 million. Now we have to remove the stage micrometer and we have to keep the slide or the object which we have to measure. For example, hibiscus flower is there, and pollen grains, we have to measure the dimensions of hibiscus pollen grains. Just prepare the slide of hibiscus pollen grains and put the slide under the microscope, which one is calibrated. Okay, now stage micrometer is not there, just ocular micrometer is there. Now from just measure the dimensions or measure the threads of ocular micrometers covering the dimension of that particular hibiscus pollen grain. Suppose the hibiscus pollen grain ranges from 30 to 40, that means 10 ocular divisions they have been there, which engulfs the hibiscus pollen grains. Then actual dimension of hibiscus pollen grain will be 10 into 6.66, which will be 66.66 million. So we will find out the actual dimensions of hibiscus pollen grains on the basis of this micrometry technique. At the B.Sc.III level, we can also give project to the students, and the students can be asked to do a project on to determine the dimensions of pollen grains of various plant species. In our college, last year, the batch of 4 students, they have performed near about 100 pollen grain dimensions of different plant species, exotic plant species. So such projects can also be given to the students, which will increase the curiosity among them related with the subject botany. Okay, so this is the actual practical related with the micrometry technique. Here, just concentration on observation table. I have explained all the principal requirement procedure in previous slide. we will observe here the observation table. In observation table, with respect to lower and higher objective, we can measure the dimension of those pollen. Again, the precaution or one thing has to be considered, the ratio or the dimension of whatever it may be, pollen, either pollen grain or fungal spores. The project can be given to students related to the fungal spores also. Different alternative spores or mucor or rhizopus spores, different species, and they will measure the dimension of different species of that particular fungus. And again, further biostat is there. With the help of biostat, they can calibrate or they can modify all those readings, and they can write either a research paper related with micrometry as well as biometry, and book also. They can write. They can write book. They can write book also on the basis of, if they perform micrometry related with the fungal 100 fungal spores, they can easily write a book also. Okay, so this can also be given as a challenging task to the students, which will be helpful to students as well as our subject also. So one thing I have to mention here, at high power and low power, the dimension should not have to be range. The dimensions of pollen grain should have to be in similar range. They should not have to vary with great exponential. Okay, so this is about micrometry technique. This is study of DNA packaging by means of micrographs. Again, this practical will there for identification and comments and describe. Here we have to provide the micrograph, and here the students should have to be known with the various terms, that is higher level of organization of genetic material within the nucleus, because the volume of genetic material greatly exceeds than the compartment in which they are present. So there must be higher level of complexity or compression among the genetic material, so that that particular genetic material will get fitted into that minimum amount of compartment, and that compartment is nothing but the nucleus. If you observe this image, from the nucleus we observe the chromosome. In chromosome at micro level we observe, we will find out nucleosome, that is higher level of compression. Then nucleosome show histoproteins H1, H2A, H2B, H3, and H4, over the which or over the octamer that DNA is wrapped. Then here we are observing the DNA. So at macro level, from nucleus, chromosome, then nucleosome, before that solenoid is there, nucleosome, and then nucleotides or DNA is there. But we have to explain all these things with the help of micrograph. And if you observe this micrograph, it is not possible to explain all the details. Okay, we will just observe that this is a thread-like or road-like structure. Google map is there, and this is nothing but a road. Okay, so students will not come to know what actually it means. Or if we just give them this micrograph, they will not get any idea related with solenoid or nucleosome. For that purpose, these pictures they have been added here. So with the help of our paper setters or senior colleagues, they will decide whether we have to give these pictures also, or we have to put only the micrograph. But 100% or definitely students will not get their concept clear by means of this micrograph. We have to provide this material also, that you have to decide. Okay, so micrograph shows solenoid model, that is further level of compression. Okay, and by means of such compression, that higher amount of genetic material, it get fitted into lower space which is there in nucleus. So again, this will be for identification and practical identification and comment purpose. We have to mention out certain specific, we have to put the entire micrograph, or we have to mention out either solenoid model or nucleosome model. At that time we have to explain the backbone around which DNA is wrapped, and that backbone is nothing but it is of histoproteins, and there are 5 different histoproteins: H1, H2A, H2B, H3, and H4. They form an octamer. Apart from H1, other remaining histoproteins, they form octamer around which that DNA get wrapped. So the question depends on you or senior colleagues, where we have to just give micrograph or we have to provide all these details to the students at the time of practice. Okay, so this is again further illustrative diagram which explains all the details, and this is colored one. When we start from the cell, we come to nucleus where genetic materials are present. They are chromosomes. Then chromosomes, we will obtain solenoid fibers. Solenoid fibers, then nucleosome and the DNA. That means the from DNA to this chromosome, there is higher level of configuration in terms of compression, and this genetic material is compressed within that particular limited space. Okay, so here we have to concentrate on nucleosome and solenoid, and we have to again concentrate the higher compression ratio of that particular genetic material which get fitted into the nucleus. So nucleosome model and solenoid model we have to concentrate during this practice. Next practical. So this is new to us. That is Beer and Lambert's law. Study of Beer and Lambert's law. Though we observe this as Beer and Lambert's law, this is nothing but a combination of two laws. First law was Beer's law, and second law is Lambert's law. So Beer's law, it states that, or it show relationship in between concentration and absorbance. Both the concentration of the medium and absorbance, absorbed light by that particular medium, they are directly proportional to each other. And second thing, that is second law, that is Lambert law. This show that absorbance and the path length, they are directly proportional. So first Lambert law was there. It states that absorbance and path length, they are directly proportional. So while put forth in this law, Lambert does not involve any medium. Lambert, irrespective of any medium, Lambert put forth that, Lambert mentioned that the absorbance and path length, they are directly proportional. But what is the difference in between Beer's law? Beer's law, or the Beer, scientist Beer, he studied the same thing, light absorbance, with related to liquid medium. So that is the difference. Okay, and one can easily remember this difference because Beer and liquid phase, again that absorbance of light related with liquid phase, Beer. Okay, so on the basis of that name, we can also remember that thing. That means Beer's law related with liquid phase. Okay, so Beer's law and Lambert's law, they have been combined together to form Beer-Lambert's law. Okay, and those Beer-Lambert law, this relates with, or this is nothing but the combination of both these law, and this states that for a given material sample, path length and concentration of the sample, they are directly proportional to the absorbance of the light. Okay, so Lambert's law show relationship in between absorbance and path length, while Beer's law show or mention the relationship between concentration and absorption. Okay, by combining all both these law, this Beer-Lambert law, it has been put forth or it has been stated, and this is nothing but the, for given material, sample path length and concentration of the sample, they are directly proportional to the absorbance of the light. Here in the diagram or in figure, we are observing that there is a column or there is a medium. Okay, and on that medium the light get incident, and that light is nothing but the, it has been mentioned by I0, that is initial light. Okay, concentrate on the size of that particular arrow. Whenever that light passes through that particular medium, we observe the same arrow with same color, but the dimensions of that particular arrow, it has been decreased. That means that denotes that whenever light passes through the medium, that light or that incident light, whenever it gets transmitted, that transmitted light, it is minimum than the incident light. That means I0 is maximum and transmitted light is minimum. Then Then what happens with the that remaining light? Some of the light it get absorbed by that medium, some of the light gets scattered, some of the light show interference, and that's why the transmitted light it is less than the I0, that means incident light. Forget about all the difficult interference, transmission, forget about all those words. Just you have to remember that the incident light it get reduced when it pass through a medium. Okay, so on the basis of this we have to perform a practical. Now the big task is there, how to perform the practical. So I will give one example, and myself will raise the objection over that particular example later on. First, I will explain the example. So here we will perform to carry out this experiment. We should have colorimeter or spectrophotometer. Okay, this is the basic requirement to carry this practical. Okay, so I will not read out all these lines. I will explain the practical with the help of table. Okay, so first I will explain the practical. Here we are observing the table. We have to prepare the known concentrations of a standard compound. For example, here potassium permanganate is there, and we will put potassium permanganate as a sample. Then we will prepare the stock solution, and we will prepare various molar concentrations of that particular stock solution by means of serial dilation. Okay, and here blank is used as distilled water or deionized water. Here we will use distilled water as a blank. So if we are using the colorimeter, what will be the scenario? And when we are using the spectrophotometer, what will be the difference to carry out this practical? But before that, just we have to prepare these test tubes as per the observation table. Test tube 1 to test tube 7. Then 0.01, this will be the stock solution. We have to add the stock solution as per the given observation table. We have to add the distilled water as per the given observation table. This will be the final concentration of that KMnO4 solution. And next column is that has not been mentioned. That column is absorbance of that particular concentrated KMnO4 solution. So what will happen? We will get the absorbance, specific absorbance on colorimeter as well as spectrophotometer. But the practical actual differs. If you use colorimeter, you have to find out the specific band or specific knob which gives maximum absorbance. Specific filter instead of knob or band, specific filter which gives maximum absorbance. So on colorimeter there are about, I think, 8 filters are there. So with the help of 1 to 8 and particular solution, we have to find out the maximum absorption showing filter. This is related with the colorimeter. Suppose the stock solution is there, we have to put that stock solution in the cuvette, and suppose filter 1 is applied, but before that filter 1 should have to be made zero by means of blank, that means distilled water, and then we have to measure the concentration of that particular stock solution. Suppose filter 1 gives minimum absorbance, and we continue the same practical up to filter 8, and suppose filter 5 gives maximum absorbance. Among all those filters, we have to choose filter 5 to carry out all the procedure, all the remaining procedures. Okay, so selection of filter is based on maximum absorbance. So here each and every time whenever you change the filter, at that time you have to make zero absorbance with the help of blank reading. Okay, so this will be in terms of colorimeter. Once again I will repeat, then we will carry this practical on colorimeter. We have to first find out the maximum absorption showing filter with respect to that particular compound, with respect to particular solution of specific compound. Okay, whenever after finding out the higher absorption filter or higher absorption, we have to choose that particular filter which the compound or that particular solution show maximum absorbance, and we have to carry further practicals on that particular filter. Okay, when the practical is going to be performed on spectrophotometer, we have to find out the specific wavelength. Instead of filter is nothing but it shows the ranges from 400 to 500, 500 to 600 in the same manner. When the practical has to be performed on spectrophotometer, we have to find out specific wavelength which show maximum absorbance related with specific concentration of that particular stock solution of specific compound. Okay, and after finding out that particular wavelength showing, suppose 420, this is the wavelength which shows highest absorption for that particular compound solution, and we have to use that 420 wavelength to carry out all the practicals. Okay, to carry out all the, or to take all the readings with respect to that particular solution compounds. Okay, now when you observe the graph, when we plot the graph against concentration on x-axis with respect to absorbance on y-axis, the graph show direct line from the point of origin. Okay, from the point of origin. Then the question arises that what is the unique thing in this practical? We can give the solution of unknown concentration. We have to give the solution of unknown concentration to the students. First, the students will plot the graph by means of known volume or known concentration of the solution. Then later we have to give unknown solution with unknown concentration, and they will find out the absorbance, and by the or with the help of that absorbance, they will find out the concentration or particular concentration of that particular unknown solution, particular concentration with respect to that unknown solution. So this is the explanation. Okay, if you have not got the concept here, you might observe this particular observation table later on. But at the beginning, stated that I will explain the details and myself will raise the question also. Now the question which has to be raised, that is why we are performing here or we are taking here KMNO4. This is the practical related with the chemistry. This is practical in chemistry, and at TY or B.Sc.II, this practical is there in physical chemistry also. So why we have to take KMNO4? Instead of these chemical compounds, we can take B.Sc.III, we can take Butyr dye, we can take Zambut Syzygium, okay, we can take Carissa Karwand, okay, and go on preparing the solutions. Again, the question is there. A big question is there. Suppose we are using the Zambut or Syzygium species, then how to prepare the molar solutions? We don't know the molarity present of the specific compounds present in that Syzygium species, B.Sc. species, Butyr species, or other dye. So here we can modify practical, or we can modify this practical by means of preparing percent solution. By means of preparing percent solution. Then how to prepare percent solution? Just just take, instead of just take Syzygium fruits, Syzygium, that means Zambut, Syzygium fruits. Then with respect to dry weight and fresh weight, we can perform this practical. Just weigh 1 gram of that fruit with respect to FW and DW, dry weight and fresh weight, and prepare 1% solution of that particular plant pigment, 1% solution of that particular fruit or seeds, or in terms of Butyr, flowers. Okay, 1%. Then that 1% solution, it can be diluted with the help of serial dilution method. Just prepare lower concentration of that 1% solution, and after preparing, ask the students to find out the absorbance. Later on, they will plot the graph, and after plotting the graph, just give the unknown sample with unknown percent. Okay, and ask the students to find out the percent value of particular pigment present in that solution. So the students will easily find out the concentration of that particular solution. Uh, this is the basic one. Again, for this practical, one should have to purchase the standards related with the anthocyanin and other pigments of the plant material. They are costing too much. So don't go for the purchase of standards to for performing this practical. Just create the interest among the students by giving various plant samples uh, related with the dye plants or plant dyes, and they will find out the concentration of that particular plant specific plant specific dyes and pigments in percent concentration. So this might be implemented, or this might be implemented at the time of examination as well as regular practical also. So one can replace the chemicals with the plant dyes also. So here, whenever we use plant dyes, at that time we have to concentrate on percent solutions. We have to make percent solutions. Okay, all the remaining procedure will be similar. So practical number, next practical, preparation of permanent cytological slides. On next slide, I have given various methods by means of which we can prepare the permanent cytological slides. These methods are acidocarmine staining, butanol schedule, and quick freeze method for the preparation of permanent cytological slides. But here I will explain the preparation of permanent cytological slides which I perform or which we use to teach the students or at the time of practical. Just prepare the solutions in the ratio of butanol with respect to acetic acid in the ratio of 25 to 75. Then go on decreasing the ratio 50-50 and 75 to 25, and last one will be the 100% butanol. So we have to decrease the percentage or concentration of acetic acid in sequential manner or stepwise manner. Just put the slide which earlier have been prepared in cytological practicals, that means the practicals related with the mitosis and meiosis. That slide has to be put, or just keep the slide in a petri dish containing these combinations of butanol and acetic acids, and at every stage the slide which has been kept in the particular medium, that slide has been kept for 5 minutes. That means every stage is for about 5 minutes. Precaution is taken, put the slide with cover slip downwards, okay, so that cover slip get removed within that particular solution, that means butanol and acetic acid solution. And later on, prepare two slides. One slide, that means covered, removed cover slip, we have to mount on fresh slide, and the slide where material, certain material, that means basic slide, we have to use Canada balsam or DPF, and again we have to mount fresh cover slip on that particular slide, and the cover slip which has been separated from the slide, it is used to prepare another slide. So at last we will observe two slides, or we will get two slides. Then observe two slides, and if you are lucky, and if you get the stages, then you are lucky. And if you get stages, this will be the permanent slide. If you do not get stages, perform the practical until you get the results, better results. So this is the basic stage, basic technique for preparation of permanent cytological slides. With the help of this technique, within half an hour, you can prepare the permanent cytological slides. I have prepared the slides related with the giant chromosomes, okay, by means of this technique. So this is the basic technique, preparation of permanent cytological slides by using acetic acid and butanol. Again, these chemicals, they are not that much costly also. Whenever you use ethanol, it requires permission and other things to purchase that ethanol or alcohol. So that's why we can modify this technique also with the help of acetic acid and butanol. So here you can later read out all these techniques, and you can perform this technique to preparation of permanent cytological slides. But this is the basic and common thing which can be concluded within 30 minutes or within half an hour, and you will get, and students also will get the permanent cytological slides, so that they will become happy also. Within 30 minutes they will get the permanent cytological slides. So these are the methods. Quick page method for making slides permanent. You can read out all those methods later on. Then this is the practical, that is study of photomicrography technique and submission of photomicrography. They are related, interrelated or correlated practicals. So I have given here the basic technique, because in most of the departments we are not with the microscope which has been fitted with the camera. So uh, at that time, anyhow, we have to complete the practical. This will not be the solution. We do not have the microscope fitted with the camera, so we have not taken this practical. So this will be not thing. We have to find out other solutions also. Okay, so this is the basic solution which I have given to you. So forget about the camera fitted with the microscope fitted with the camera. Forget about all the things. We have forget about the laptop and computer that is needed for the printing purpose. Okay, we have compound microscope. Majority of the students, they have smartphones. Ask the students to take the photograph with the help of their smartphones. Again, nowadays there are equipments easily available in the markets also. Here you can, in this image we are observing that the microscope or the smartphone, it can be easily connected. It can be easily connected to the microscope by means of an adapter. Okay, so this microscope, this is completely equipped with the camera, but we are assuming that we do not have this microscope fitted with the camera, and so that's why we are performing the practical. If the department, any department, if it has such instrument, then well and good. But if there is no such instrument, we can perform this practical also. With the help of smartphone, just capture the image. After capturing the image, again the question arises, we do not have Adobe Photoshop, other materials or other softwares, they are again costly. Forget about all those softwares. Just go to the Microsoft PowerPoint. Here, transfer that image which has been taken in your smartphone, and after transferring the image, just edit the image with the in the Microsoft PowerPoint application, label the image, and make a printout of that photo micrograph. Again, nowadays, if you search Google Play Store, we will find out n number of applications which are related with the photo editing and photo labeling. So with the help of those applications also, we can ask the students to edit and label those photographs. We can give homework. We can give homework to the students also. Just capture the image and edit and label the photographs at your home, and bring those edited and labeled photographs in department on next day, and just take out the printouts. So this is the basic. If any department has highly equipped microscope, then well and good. Just you have to keep the slide and put the button. Okay, that automatic, that photograph will be there on your computer or laptop screen. So I have put the this practical to those departments, they which do not have such high highly equipment, that means microscope with fitted with the camera. So next slide, these are various applications which are present on Google Play Stores, can be easily downloaded, freely downloaded on our smartphone. And just do not download all those applications in our smartphone. Ask the students to download to them, and ask the students to edit the photographs or the captured images, and then ask the students to label also, and just take out the print of that particular photograph. So again, there is submission. The practical is for submission also. Students have to submit at least 10 or 20 different photomicrographs, and they will they have to again submit those that file at the time of exam. That will depend as per the nature of practical examination. Okay, so here I conclude my presentation. Lastly, I am very much thankful to all the participants and the organizing committee members of this workshop. I'm very much thankful to Dr. Anil Jagtap sir for inviting me as a resource person. This is the first time I am speaking as a resource person. So I'm very much thankful to Anil Jagtap sir, and I hope so that the scenario related with me at last should not be there. कभी कभी हम जी को यूं ही बहलाते हैं जो खुद ना समझ सके दूसरों को समझाते हैं. So this would not be the scenario with respect to me. I am very much thankful to all the audience also. So thank you very much.
Thank you very much, sir. I request participants to raise their hands or ask questions. Is there any question? Also, participants can paste their questions in the chat box. After the presentation of resource persons, they will use to answer these questions. Is there any question, please? Looking, there is no question from this presentation. Dr. S.A. Deshmukh sir, you have keenly elaborated all the practicals with the effective and realistic and some realizing slides, and very kind suggestions you have given and guided to us and participants. We have Dr. E.B. Bhalerao sir, in charge principal of Kisanveer Mahavidyalaya, Y, or as a research, as a chairperson for this session. I would like to request Dr. E.B. Bhalerao sir to put his remark on this presentation. Please, sir.
Good afternoon to everybody. May I audible to everyone?
Yes, sir.
First of all, I am thankful to the University Grants Commission for sanctioning this grant for this particular webinar cum seminar. Now here, I am also proud of Dr. V.V. Kamble, who has organized this webinar, and also I am also thankful to the principal of the Daiwadi College, Daiwadi, who have granted the permission to conduct the same. I am also here thankful to the today's resource person, Dr. Sagar Deshmukh, who have reviewed all the practicals of this newly included syllabus, and he also covered the practicals showing the procedures or the protocols, the nice slides he had here shown to us. One more thing I want to notice here, that the steps, whatever the steps taken for the in the procedure in the mitosis and even the meiosis, along with slides, nicely presented, sir. And on behalf of all these here the participants, I appreciate your presentation. And regarding the DNA packaging, you have also given the nice information. Then a new practical which have been incorporated or included in this syllabus, that is Beer's law, study of Beer and Lambert's law, nicely explained, sir. In conclusion, you have given nice presentation which will be useful for each and every participants. Once again, thanks for giving me this opportunity. Thanks to everybody.
Thank you, sir. Thank you very much for being with us in spite of your busy schedule. Now, I would like to request Miss P.R. Nale, madam, to propose a vote of thanks. Please. Just a minute, sir. Just a minute, sir. Madam. हाँ मॅडम झाले अनम्यूट अनम्यूट झाले का
Yes. Very good. Myself, Padmanari. I am very glad to propose a vote of thanks for a first session of a day. First of all, I would like to propose a hearty vote of Dr. S.A. Deshmukh from Department of Botany, the New College, Kolhapur, for giving his valuable guidance and remark on practical third based on paper 11 and 16. He explained practicals in details. We also thankful to chairperson or chairing this session, Dr. E.B. Bhalerao. Thank you, sir. Thank you for your valuable remark. Please, Mrs. Kamble, sir.
Thank you, madam. We have second resource person for this session, Dr. S.Y. Jadhav, sir. He is working as a head, Department of Botany, Yashwantrao Chavan, Varna Mahavidyalay, Varna Nagar. He has completed MSc, MPhil, PhD degree, also qualified a SET exam. He has 7 years of teaching at PG level and 16 of at UG level. He has 5 years of industrial experience. He has published 9 research papers and presented 16 research papers. He has attended 40 seminars and workshops. He has worked as a resource person for 4 workshops. His research areas are tissue culture of medicinal and horticultural plants, cultivation practices of medicinal plants, standardization of herbo-mineral preparations. He guided to several projects in environmental sciences, agrochemical and pest management, sericulture, chemical engineering, and pharmacy. We have chairperson Mr. Samson Gaikwad, sir, Head Department of Botany, Yashwantrao Chavan Institute of Science, Satara. With the kind permission of chairperson, I request Dr. S.Y. Jadhav, sir, to deliver his presentation based on practical 3 again. Please, sir.
चालू करायचं चाललंय आता त्याचं हे झालं vote of thanks.
Hello. Hello.
Yes, sir.
Am I audible to you, everybody?
Yes, sir. Yes, you are audible.
Screen allowed screen sharing.
Just I'm sharing screen with all of you.
Allowed.
Please allow us to share the screen. The share.
I will make, sir.
Share the screen.
Okay.
Yeah.
You can share, sir, now.
Yeah.
हो हो
आता हो बरोबर. मी केलाय की आता ते मेटल स्टडी केलाच की आम्ही रोड साइड प्लॅनचा.
Hello, dear
Yes.
friends. We are going to discuss on practical 4, of which many practicals are quite new to the botany students. As a botany student, Bhushnav sir, 2 मिनिट थांबा की जरा. पुढं कसं जायचं? पुढं कसं जायचं म्हणजे? हां.
खाली होते.
बरं. हां ठीक.
आलो 2 मिनिट.
So many practicals are quite new in this area. So first practical is biofertilizers. Here I have defined what are the biofertilizers and what are their applications. So there are many bacteria and algae which are provided micronutrients to the plants, and there are various bacteria like rhizobium. It is one of the bacterium which is present in the root nodules. Then azospirillum, which is again nitrogen then symbiotic nitrogen fixing bacteria, that is cyanobacteria, nostoc. Then this is the picture of nostoc. Then free living nitrogen fixing bacteria, rhizobium azospirillum, trichoderma, that is the fungus which is used as a biopesticide to get a remedy of various diseases. Then azotobacter, these are also nitrogen fixing bacteria which fix about 78% nitrogen. PSB, then important biofertilizers, applications of biofertilizers, shielding tip. Then main field of application. So how to use the biofertilizers, that is the importance of that. Then how to apply biofertilizers, at what rate. So 4 to 5 kg of azotobacter biofertilizers are generally used. So here is the chart given, which shows for which crop which type of bacterium is recommended and in what proportion. So here you can see all the chart. In fact, I am going to share all my PPTs to all of you through regarding using biofertilizers. So 1 into 10 raised to 8 cells per ml should be present in the bacterial preparation. Then this method is applicable to rice crops. The seedlings are planted in the bed of water for 8 to 10 hours. Then seed treatment, that is also recommended for microbial biofertilizers. Then
ग्राउंड प्रायव्हेट हेलिपॅड वापरायचं काहीतरी होतं ते मला कन्फ्युज झालं नेमकं काय करायचं.
next hello.
Hello.
Just a minute, sir. Hello. There is some technical problem.
Hello, sir.
Just a
Hello,
minute,
sir.
sir.
You are audible.
Yeah. Then next PPT, that is the crotalaria as a biofertilizer. So here you are going to see the plant of crotalaria. So use of crotalaria as a biofertilizer. So let us see the classification of this crotalaria: kingdom plantis, subkingdom phanerogams, deviant dicotyledony. So all the classification details have been given here. Then introduction, as all of we know, this plant is used as a biofertilizer for many years. It is also called as brown hemp or Indian hemp, and its morphology is it is a annual herb, and having a 1.2 to 2.5 100 to 1 meter tall plant. Then leaves are simple. Then we have described here the morphological characters of that plant. Then part used, we can use whole plant in fresh or dry condition. So here are different uses of crotalaria. So what are the different uses of crotalaria? Here we can see. So it is used as a green manure. Then it is also nitrogen fixing legume, and so it has use. So it's a so here is another practical decoction of Adathoda vesica or Adathoda zelenica, family Acanthaceae adulta, part used fresh or dried leaves, flower, bark, or root. It is a very good anthelmintic plant, and it relieves cough from different part plant here on doshas. In Ayurveda, Vata, Pitta, and Kapha are three different doshas which have action on the body. So active principles of adaptation have been given here in this slide. Then preparation of decoction garden. Wash the leaf to remove any dirt from the surface with the tap water. Weigh 10 grams of leaf and cut it into small pieces. So whole procedure has been given here. Then properties of Vasaka decoction. So health benefits, there are various health benefits which have been described in this slide. So I'm going to share all. Food adulteration. Then next practical is the food adulteration, in which we are going to analyze different adulterated things which are sold in market, and we are not knowing about much more things about adulteration food. So saffron, as we know, saffron is the very costly commodity which is used. Dried stigma of Crocus sativus, that is used as a saffron. So FDA and ISO specifications have been given for saffron. Recently, we have read a news in the newspaper that in Mahabaleshwar there is successful plantation of saffron. So how to make a difference between genuine saffron? So saffron is admixed with the some colors, or it may be added with maize comb, or ज्याला आपण मक्याच्या कणसाचं शेंडे म्हणतात, तुरे म्हणतो, ते तुरे त्यात वापरले जातात. याच्यामध्ये ट्रॅडिशनल हिलर्स they trust in pharmaceutical honkos cover it. Scientific confirmation about turmeric extract. आता टर्मरिकचा आपण बोलतोय, कर्कुमा लवंगा, ह्याच्यामध्ये पण अडल्ट्रेशन दिसू शकतं आपल्याला. टर्मरिक इज अ फॅन्टास्टिक नॅचरल येलो कलर डाय, आणि the Indian Standards Bureau has set the minimum limit of curcumin content 3% in curcumin. Adulteration in turmeric powder is prevent anywhere in market which robbed this golden spice for its true use. How to detect the Adulteration turmeric. Add a small quantity of turmeric in a whole transparent glass of water. Pure turmeric will not leave any color. Adulterated turmeric appears to be bright in color and leaves a color immediately in water. So add a teaspoon of turmeric powder in a glass of water. Natural turmeric powder leaves light yellow color while settling down. Adulterated turmeric powder will leave a strong yellow color in water while settling down. Mix few drops of iodine to turmeric solution. If blue color is present, in that, then it is known as Davidson because it has got very bad, and it is also added that it has been listed by me that the plants of this asafoetida traders identify the asafoetida, burn small quantity of asafoetida in a stainless steel pool. Pure asafoetida will burn like camphor. Adulterated asafoetida will not produce bright flame like camphor. Mix few drops of iodine to the asafoetida solution. If blue color appears, starch is present. Shake little portion of the sample with water, soapstone or other earthy material at the bottom. If asafoetida is adulterated, soapstone or other earthy matter is present. Then adulteration for camphor. So camphor, it is a religious commodity in India. We are using in different pujas. Basically, camphor is extracted from plant Cinnamomum camphora, but nowadays synthetic camphor is also available. So alpha pinene and beta pinene are used for this purpose, which are showing properties like that of camphor. Then Osimum kilimind, so memplability, which give camphor, and addition in this, the material shall consist of crystals, granules, or crystal mass of tough consistency and pulverulent. Character nose flower of camphor having pungent aromatic taste, followed by sensation of cold and characteristic pungent penetrating odor. Technical material may also be in the form of powder tablets or any other form. So these are some of the simple methods of identifying the adulteration in the camphor, as well as solubility is concerned. Camphor can be also dissolved in water or in chloroform, and it shows that these are some of adulterants from the camphor. Then next slide is organoleptic. So next practical is the organoleptic characters of the plants. So Ayurveda got long history in, and it has been used since 4,000 years. And according to Ayurveda, the organoleptic characters are one of the important tools
Please share the PPT, sir.
Yes.
Yes, it is visible now.
So there are four plants for organoleptic characters. First one is Ocimum santum, that tulsi, family Lamiaceae. So when a dried stem is observed under by visual eyes, then it is a herbaceous plant having height of 56 centimeter, much branched annual herb found throughout the country. Stem is wiry, branched, hairy, soft, blackish brown externally and pale violet internally. It is branched, subquadrangular, black internally, cream color. थोडक्यात ऑर्गॅनिक कॅरेक्टर म्हणजे रंग रूप वास चव ह्याची माहिती त्या प्लांट मटेरियलची घेणं, तो आर दी ऑर्गॅनोलेप्टिक कॅरेक्टर्स. पुढचा प्लांट आहे जिंजर, जिंजिबर ऑफिसिनालिस. ह्याला आपण आलं किंवा अद्रक म्हणतो, पण आयुर्वेदामध्ये जास्त करून सुंठ वापरतात. आपल्याकडे आल्याचे दर बऱ्याच वेळा पडलेले असतात, परंतु सुंठ मात्र 250-300 रुपये किलो या दराने विकली जाते. फक्त आल्याची सुंठ कशी करायची आपल्याला माहित नाही, तर त्यासाठी थोडंफार आपण प्रयत्न केले पाहिजे. तर ह्याच्यामध्ये जिंजर मध्ये रायझोम्स असतात, विच आर डगड इन जानेवारी टू फेब्रुवारी, आणि त्या रायझोम वरती आपल्याला बर्ड्स आणि रूट्स त्याच्यावर दिसतात. ड्रग ऑकर्स इन एंटायर रायझोम ऑर इन पीसेस. हे अखंड रायझोम किंवा रायझोम तुकडे असतात. रायझोम इज लॅटरली कॉम्प्रेस्ड, bearing flattish ovate branches on upper side, each having depressed scar at its apex. Pieces are 5 to 15 centimeter long, 1.5 to 6.5 centimeter wide, and 1.1 to 1.5 centimeter thick. Fracture short with projecting fibers. Transversely cut surface shows a wide central stele having numerous grayish cut ends. Odor is gingery and taste is pungent. Then Avla, Imlica officinalis, family Euphorbiaceae. It is Indian gooseberry. Generally, in Ayurveda, dried fruit pericarp of this fruit is used. It is mostly collected in winter after ripening. The drug consists of curled pieces of pericarp of dried fruit occurring either as separated single segment 1 to 2 centimeter long or united as 3 or 4 segments. Bulb color is gray to black, pieces showing broad, highly shriveled and wrinkled external convex surface to somewhat extent. Transversely wrinkled lateral surface, external surface shows a few whitish specks. Occasionally, some pieces show a portion of stony testa. So these are some of the characters shown by the Avla. Then next PPT.
संजय झालो आहे. माझा दोस्त आहे.
सगळ्यांचा दोस्त आहे मी. हॅलो रम्या.
हॅलो सर.
हॅलो 2 मिनिटं.
हॅलो सर.
हा 2 मिनिटं.
हा मला बोलायलाच काही ना सर.
So next practice is preparation of hair oil from Eclipta alba or Bhrungraj for hair growth. आपल्याला माक्याचं सगळ्यांना झाड माहिती आहे, फॅमिली ॲस्ट्रेसी. त्याला भृंगराज असं म्हणतात. इंग्लिश नेम ट्रेलिंग इक्लिप्टा प्लांट, ट्रेड नेम भांगरा. पार्ट युज्ड होल प्लांट, त्याचा अखंड प्लांट वापरतात. ह्याच्या पण मेडिसिनल व्हॅल्यू बऱ्याच आहेत. आपल्याला माहिती आहे की भारतात जगामध्ये अजून 80% पॉप्युलेशन आयुर्वेदिक मेडिसिनवरती अवलंबून आहे, आणि याच्यामध्ये बरीचशाच वनस्पती वापरल्या जातात. आपल्याकडे भारतामध्ये माक्याबरोबरच ॲलोव्हेरा, इम्लिका ऑफिसिनालिस, हिबिस्कस रोझासायनेन्सिस किंवा रिठा, शिककाई, मेहंदी यासारख्या वनस्पती या तेल बनवण्यासाठी वापरतात. त्यात माक्याचं तेल हे जरा प्रसिद्ध आहे. आपण डाबर आवला केस तेल ते बघितलं, त्यामध्ये माका वापरला जातो. आवळ्याचं पण तेल असतं त्यामध्ये. मॉर्फॉलॉजिकल कॅरेक्टर्स आपण दिलेली आहेत. त्यामध्ये लिव्स सिंपल आहेत, अपोजिट आहेत, ससाईल आहेत, टू सब ससाईल. प्लांटची उंची जी आहे ती 30 ते 40 सेंटीमीटर असते. 1.5 ते 1.9 सेंटीमीटर वाईड ऑबलॉंग लॅन्सी रोट लिव्ज असतात. फ्लावरचे कॅरेक्टर त्यात दिलेले आहेत. फ्लावर्स आर व्हाईट इन कलर. Bracts are about 8 in number. The corolla is 4-toothed. Stamens 5. Filament epipetalous free. Anther united into tube with base abscess. So these are some floral characteristics. Climate and soil. This plant can be grown in sandy to clay soil. आणि जनरली आपल्याला हे प्लांट गटराच्या कडेला उगवलेलं बघायला मिळतं किंवा मॉइस्ट प्लेसेस मध्ये बघायला मिळतं. ह्याचं प्रपोगेशन जे आहे ते सीड न करतात किंवा स्टेम कटिंग करतात. ह्याचं तेल कसं करायचं तर साधारणतः 1 किलो माका घ्यायचा. हा फ्रेश किंवा ड्राय असायला चालतो. त्यामध्ये अलाउ टू सोक इट इन 4 liter of water for overnight in a pot. Boil the decoction until the fraction reduces to 1 liter. Filter the decoction through strainer. It is reddish in color. लक्षात ठेवा माक्याचा काढा हा लालसर रंगाचा दिसतो. ऍड 1 liter of sesame oil or coconut oil to the filtrate. ह्याच्यामध्ये आपल्याला 1 liter sesame oil किंवा coconut oil घालायचं. हे अगेन पुन्हा बॉईल करायचं until water gets completely evolved आणि फक्त त्यामध्ये तेल राहिलं पाहिजे. Allow to cool the oil and again filter it through strainer. Store in a dry clean bottle for further use. You can change the proportion in accordance to your practical purpose. सो ऑइल कसं दिसतं तर थिक ब्लॅक लिक्विड आहे. स्ट्रॉंग अरोमॅटिक ओडर आहे. कलर त्याचा ब्लॅकिश आहे. प्लांट पार्ट इज द लीव्स ऑर होल प्लांट. आणि एक्स्ट्रॅक्शन मेथड स्टीम डिस्टिलेशन किंवा वॉटर डिस्टिलेशन. ह्याच्यामध्ये युजेस काय काय आहेत तर इक्लिप्टा अल्बा ऑइल डायरेक्टली नरिशेस अँड इम्प्रूव्ह द हेअर ग्रोथ. इक्लिप्टा अल्बा ऑइल इज अ गुड अँटी डॅन्ड्रफ ऑइल. Eclipta alba oil for hair growth is also good for pure mature graying for hair. Then Eclipta alba oil is good for skin care too, as it is good anti-aging product. पुढची स्लाइड. नंतर पुढचं प्रॅक्टिकल जे आहे ते आपण बघूया. प्रिपरेशन ऑफ शॅम्पू फ्रॉम सोपनट. सोपनट म्हणजे रिठा. तर रिठ्यापासून आपण शॅम्पू कसा बनवू शकतो हे आपण यामध्ये बघूया. सोपनट्स आर द फ्रूट दॅट ग्रो ऑन श्रब्स इन ट्रॉपिकल रिजन्स ऑफ आफ्रिका, आशिया, युरोप अँड अमेरिका. आता ह्याच्यामध्ये रिठ्याचं सायंटिफिक नाव जे आहे ते सॅपिंडस लाउरिफोलियस किंवा सॅपिंडस ट्रायफोलियस असं आहे. आणि याच्यामध्ये जर का आपण हे फ्रूट चुरगाळून बघितलं ओलं फ्रूट तर त्यामध्ये सोपी असं एक लिक्विड असतं. जर का आपण ते पाण्याखाली दिलं तर त्याचा फेस भरपूर येतो. त्यामुळे रिठाचा शॅम्पू करणं हे अत्यंत सोपं आहे आणि हा शॅम्पूचे उपयोग पण फार चांगले आहेत. त्यामध्ये आपण बघू शकतो की हाऊ टू प्रिपेअर शॅम्पू. त्यासाठी रिक्वायरमेंट काय काय आहेत. आपल्याला रिठे लागणार आहेत, प्लेन वॉटर लागणार आहे, मिक्सर ग्राइंडर लागणार आहे, हॉट प्लेट, मस्लिन क्लॉथ आणि बीकर्स. इफ यू हॅव 10 soapnuts, place them in a small muslin cloth. Place medium saucepan with 2 cups of the water and bring to boil. Reduce the heat and smear for 20 minutes. Add 1 cup of water and simmer for 10 more minutes. Remove from heat and let it cool. Squeeze out the bag until we get whole juice from the fruits. The soap nuts are also utilized to create psoriasis and eczema, to cure psoriasis and eczema. So and it also soothes body. It's working as a deep cleaning natural shampoos. And if you are going to make a large batch of soap nut shampoo bars, you can store the solution in an ice cube tray, then freeze them so you can use that shampoo anytime. So what are the other different plants from which shampoo can be prepared? So preparation of shampoo from shikakai. So Acacia consina is the name of this plant. We know that the pods of this plant, those are used as surfactants or a cleansing agent. So the shikakai is rich in vitamin C and many essential phytonutrients that nourishes the hair follicles and promote the hair growth. Shikakai contains natural surfactants called saponins, which makes a great natural cleanser. आणि शिकाकाईचे बरेचसे उपयोग आपण इथे बघतोय. शिकाकाई is an excellent alternative for people who are allergic to sulfates and parabens in the commercial shampoo. The natural saponins in shikakai not only cleans hair, they add shine and body and make the hair look thicker and smoother. So requirement, what are the requirement? Shikakai pots is the main requirement, plain water, mixer grinder, hot plate and beakers. And then take 10 grams of shikakai, about 5 to 6 shikakai pots. Take all the ingredients in a pan, add 750 ml of water and let them soak for about 8 hours. Or you can keep this skip this step and straight away make the shampoo. Heat the mix till it starts to boil. Reduce the flame slightly and let it boil for 5 to 15 minutes. The longer you cook, the more concentrated it will become. Add more water if you wish. Remove from the heat, cover and let it cool. When it comes to the room temperature, crush the shikakai with your hand. Strain before use. Use normal shampoo. Use as a normal shampoo. जनरली आपल्याकडे माहिती आहे की आपल्या हिंदुस्थानामध्ये किंवा भारतामध्ये लेडीज ज्या आहेत ह्या शिकाकाईचं पाण्याने आंघोळ करतात, कारण त्यामुळं केसाची वाढ चांगली होते. आणि दिस शॅम्पू इट सूट्स ऑल टाइप्स ऑफ हेअर्स, सगळ्या प्रकारच्या केसावरती याचा चांगला उपयोग होऊ शकतो. देन नेक्स्ट प्रॅक्टिकल इज द yes क्वालिटेटिव्ह टेस्ट फॉर अल्कलॉइड्स. आता इथनं पुढची सगळी प्रॅक्टिकल आहेत ही बऱ्यापैकी प्लांट मध्ये डिफरंट केमिकल कंटेंट्स आहेत त्याच्यावरती अवलंबून असणार आहे. ह्याच्यामध्ये आपण एम, प्रिन्सिपल, रिक्वायरमेंट आणि मेथडॉलॉजी आणि त्याचं टेस्टिंग मेथड्स हे आपण या थोडक्यात सगळे दिलेले आहेत. यामध्ये पहिला आहे तो अल्कलॉइड. अल्कलॉइड्स दे आर बेसिक and can accept hydrogen ions from other compounds. Most alkaloids are isolated from plants but have medicinal uses in humans. Alkaloids are a class of basic naturally occurring organic compounds that contain at least one nitrogen atom. So they can be qualitatively estimated by following method. ह्या सगळ्यामध्ये आपण बघतोय की ज्यामध्ये सॉक्सेट अपॅरेटस नावाचं अपॅरेटस दिलेलं आहे. हे जर का आपल्याकडे नसेल तर आपण साधं डिस्टिलेशन पण करू शकतो आणि मटेरियल मिळवू शकतो. त्यामध्ये केमिकल जी लागणार आहेत ती इथेनॉल, इथिल एस्टेट, एन-हेक्झेन, कॉन्सन्ट्रेटेड हायड्रोक्लोरिक ऍसिड आणि मेयर्स रिएजंट. ह्याची प्रोसिजर पण पूर्णपणे दिलेली आहे. एक्स्ट्रॅक्ट कसा करायचा आणि हाऊ टू टेस्ट दॅट एक्स्ट्रॅक्ट. त्याच्यामध्ये इथेनॉल एक्स्ट्रॅक्ट आहे, इथिल एस्टेट एक्स्ट्रॅक्ट आहे, ऍक्टिस एक्स्ट्रॅक्ट आहे आणि एन-हेक्झेन एक्स्ट्रॅक्ट. तर ह्याच्यामध्ये आपण बघितलं तर in a clean and dry test tube, take to then add few drops of Mayer's reagent. Presence of green color or white precipitate indicates the presence of alkaloids. So that is the testing method for this alkaloids. Next. Next practical is qualitative test for flavonoids. So flavonoids, it is again important constituents from the plant. So these are the actually water soluble compounds such as anthocyanin and flavones that includes pigments ranging from color from yellow to red to blue and occur especially in fruits, vegetables and herbs. तर फ्लेवोनॉइड्स हे अत्यंत महत्वाचं कॉन्स्टिट्यूशन आहे प्लांट मधलं. ते पण क्वालिटीवर आपण इस्टिमेट करू शकतो. ह्याच्यासाठी पण आपण जे रिक्वायरमेंट दिलेले आहेत त्यामध्ये टेस्ट ट्यूब, सॉक्सेट अपॅरेटस, ग्लास बॉटल, टेस्ट ट्यूब, होल्डर, बर्नर, ऍब्सॉर्ब, ॲझोस्टॉस सीड्स, वायर गॉज हे सारखं वापरतो. आणि केमिकल मध्ये इथेनॉल, इथिल एस्टेट, एन-हेक्झेन, दोन नॉर्मल सोडियम हायड्रॉक्साईड हे यूज करायचं. त्याच्यामधली प्रोसिजर जी आहे त्यामध्ये इन अ क्लीन अँड ड्राय टेस्ट ट्यूब टेक 2 ml ऑफ एक्स्ट्रॅक्ट. टू दिस ऍड 1 ml ऑफ 2 normal sodium hydroxide. Then presence of yellow color indicates presence of flavonoids. So केमिकल जे की सगळी सांडासारखी केमिकल आहेत आणि आपण सगळ्या प्रॅक्टिकली वापरतो. या टेबल मध्ये आपण बघतोय की त्याचं फ्लेवोनॉइड्स आहेत किंवा नाही आहेत हे आपण इथेनॉल, इथिल एस्टेट आणि ऍक्टिस एक्स्ट्रॅक्ट मध्ये बघतोय. शिवाय एन-हेक्झेन एक्स्ट्रॅक्ट मध्ये पण बघतोय. पुढचं प्रॅक्टिकल qualitative test for saponins. सो क्वालिटेटिव्ह टेस्ट फॉर सॅपोनिन्स, त्यामध्ये आपण बघतो की वॉटर सॅपोनिन्स, एनी ऑफ द व्हेरियस मोस्टली टॉक्सिक ग्लुकोसाइड्स दॅट ऑकर इन प्लांट्स, they are characterized by their property of producing soapy leather, especially hygroscopic amorphous saponin mixture used specially as a foaming and emulsifying agent. थोडक्यात साबणासारखा प्रकार आहे. आणि याच्यामध्ये पण आपण ॲपॅरेटस बघितली तर 30 ॲपॅरेटस आहेत. नंतर केमिकल मध्ये बघितलं तर इथेनॉल, इथिल ॲसिटेट, एन हेक्झॅन, डिस्टिल वॉटर यासारखी केमिकल त्यामध्ये वापरतोय. प्रोसिजर ही तसं दिलेली आहे. आणि हाऊ टू टेस्ट दॅट plant material for the saponins that we can see here. In a clean and dry test tube, take 2 ml of extract and to this add 2 ml of distilled water. Shake it properly in graduated cylinder for 15 minutes lengthwise. It results in formation of 1 centimeter layer of foam that indicates the presence of saponins. So इथं आपण पुन्हा टेबल दिलेला आहे. या टेबल मध्ये आपण बघतोय की इथेनॉल एक्स्ट्रॅक्ट, इथिल ॲसिड एक्स्ट्रॅक्ट, ॲक्विस एक्स्ट्रॅक्ट आणि एन हेक्झॅन एक्स्ट्रॅक्ट त्यामध्ये सॅपोनिन आहेत किंवा नाही आहेत ते आपण सांगू शकतो. पुढचंच आहे. देन क्वालिटेटिव्ह अनालिसिस फॉर स्टिरॉइड्स, त्याच्यामध्ये आपण बघू शकतो की स्टिरॉइड ही अत्यंत तशी डेंजरस मानली जातात. स्पेशली ज्यांना संधिवात वगैरे आहेत ह्याच्यामध्ये बरीच गावठी औषधे विकतात, त्यामध्ये स्टिरॉइड्स घातलेले असतात. हे त्या औषधामध्ये स्टिरॉइड आहे का नाही कसं ओळखायचं, तर दीज आर दी लार्ज स्केल ऑफ ऑर्गॅनिक कंपाऊंड्स विथ कॅरेक्टरिस्टिक मॉलिक्युलर स्ट्रक्चर कंटेनिंग 4 रिंग्स ऑफ कार्बन ॲटम्स. याच्यामध्ये इन्क्लूड एनी हार्मोन्स, अल्कलॉइड्स, विटामिन्स. स्टिरॉइड्स आर इम्पॉर्टंट इन बायोलॉजी, केमिस्ट्री, मेडिसिन, एनी लार्ज ग्रुप ऑफ फॅट सोल्युबल ऑर्गॅनिक कंपाऊंड्स as the steroids, bile acids and sex hormones, most of which have specific physiological action. ते पण आपण क्वालिटेटिव्हली एस्टिमेट करू शकतो. ह्याच्यामध्ये केमिकल पुन्हा लागणार आहे इथेनॉल, इथिल ॲसिटेट, एन हेक्झॅन, क्लोरोफॉर्म, कॉन्सन्ट्रेटेड सल्फ्युरिक ऍसिड आणि डिस्टिल वॉटर. ह्याच्यामध्ये पण आपण बघतोय एक्स्ट्रॅक्शन प्रोसिजर पण दिलेली आहे. मगाशी म्हटलं तुमच्याकडं सॉक्सलेट ॲपरेटर नसेल तर साधं डिस्टिलेशन असं देखील चालू शकते. तर ह्याच्यामध्ये हाऊ टू टेस्ट फॉर द स्टिरॉइड्स. In a clean and dry test tube, take 1 ml of extract and equal volume of chloroform. Add few drops of concentrated sulfuric acid to it. Appearance of brown ring indicates the presence of steroids, and appearance of bluish brown ring indicates presence of phytosteroids. So this is the table for comparative presence of steroids: ethanol extract, ethyl acetate extract, aqueous extract and N-hexane extract. आणि त्याच्यामध्ये आपण कन्क्लुजन खालती मांडू शकतो. पुढचं प्रॅक्टिकल बघूया आपण. क्वालिटेटिव्ह अनालिसिस ऑफ टॅनिन्स. ह्याच्यामध्ये आपण बघू शकतो की टॅनिन जे आहेत these are the yellowish or brownish bitter tasting organic substance present in some barks and other plant tissues consisting of derivatives of gallic acid. Most of the tannins present in tea are antioxidants that help to fight activities like fight cavities, diarrhea and some even prevent हार्ट डिसीजेस. ह्याच्यामध्ये पण आपण बघतोय की केमिकल कुठलं लागणार आहे. त्यामध्ये आपल्याला इथेनॉल लागणार आहे, इथिल ॲसिटेट, एन हेक्झेन आणि 5% फेरिक क्लोराईड सोल्युशन. त्यामध्ये हाऊ टू प्रिपेअर एक्स्ट्रॅक्ट. सो मगाशी म्हटलं तर पुन्हा इथे आपण डिस्टिलेशन असेंब्ली वापरू शकतो. आणि ह्याच्यामध्ये हाऊ टू टेस्ट इट. टेक 1 ml ऑफ एक्स्ट्रॅक्ट इन अ टेस्ट ट्यूब अँड ऍड टू दिस 2 ml ऑफ 5% फेरिक क्लोराईड. फॉर्मेशन ऑफ डार्क ब्ल्यू और ग्रेनिश ब्लॅक indicates presence of tannins. जर का आपल्याला ह्या रंगाचा कलर मिळाला तर त्यामध्ये टॅनिन्स प्रेझेंट आहेत असं आपण म्हणू शकतो. या इथं पुरा कंपॅरेटिव्ह टेबल दिला आहे. इथेनॉल एक्स्ट्रॅक्ट, इथिल ॲसिटेट एक्स्ट्रॅक्ट, ॲक्विस एक्स्ट्रॅक्ट आणि एन हेक्झेन एक्स्ट्रॅक्ट. पुढचं प्रॅक्टिकल त्यामध्ये आपण बघूया आता. qualitative analysis of terpenoids. Terpenes are the compounds which are derived from plants, especially from the non-oxygen containing group. Terpenoids are widely found in plants and can form cyclic structures such as sterols. In plants, terpenes and terpenoids are important mediators of ecological interaction. For example, they play a role in plant defense against herbivory, disease resistance, attraction of mutualists such as pollinators, as well as potentially plant communication. They can qualitatively estimated as follows. त्यासाठी पण केमिकल लागणार ते आपल्याला इथेनॉल, इथिल एस्टेट, एन सल्फ्युरिक ऍसिड आणि क्लोरोफॉर्म लागणार आहे. ह्याच्यामध्ये पण आपण एक्सट्रॅक्शनची प्रोसिजर पूर्ण दिलेली आहे आणि ह्याच्यामध्ये हाऊ टू टेस्ट फॉर टर्पिनॉइड्स इन अ क्लीन ड्राय टेस्ट ट्यूब टेक 0.5 ml of the extract. It is treated with 2 ml chloroform and add concentrated sulfuric acid. Formation of red brown color at interface indicates the presence of terpenoids. तर याच्यामध्ये आपण पूर्ण प्रोसेस दिलेली आहे आणि टेस्टिंग मेथड पण दिलेली आहे आणि इथं खालती आपण त्याचा प्रोटोकॉल दिला आहे टेस्टिंगचा आणि हाऊ टू राईट दी कन्क्लुजन्स ऑफ द सेम. पुढचं प्रॅक्टिकल आहे. पुढचं आहे टर्पिनॉइड झालं. रिड्युसिंग शुगर्स. Next practical is a qualitative test for reducing sugar from given material. Reducing sugar is any sugar that is capable of acting as a reducing agent because it has free aldehyde group or free ketone group. All monosaccharides are reducing sugars along with some disaccharides, oligosaccharides, and polysaccharides. Reducing sugar can be oxidized by weak oxidizing agents. In some medium, reducing sugars generate one more compounds containing an aldehyde group. This is characteristic property of reducing sugar. Examples of reducing sugar include glucose, fructose, galactose, and monosaccharides. ह्याच्यामध्ये पण आपण काय काय केमिकल लागणार ती दिलेली आहेत आणि एक्सट्रॅक्शन मेथड पण दिलेली आहे. ह्याच्यामध्ये पेंटाहायड्रेट किंवा कॉपर सल्फेट CuSO4 5H2O 100 grams of sodium carbonate and 173 grams of sodium citrate in distilled water. हे सगळं मिक्स केल्यानंतर आपल्याला मिळतो बारकोड रिएजंट. हा बारकोड रिएजंट आपण टेस्टिंगसाठी वापरणार आहे. प्लांट एक्सट्रॅक्टची प्रोसिजर आपण दिलेली आहे. बारकोड टेस्ट इज used to detect the presence of monosaccharide in a solution. Barcode reagent is a mixture of acetic acid and copper second acetate is combined with the test solution and boiled. A red copper oxide precipitate is formed which indicates presence of reducing sugar. Or in a clean dry test tube, take 2 ml of extract. To this add Benedict's reagent. Initially we get aqua blue color of mixture. Heat mixture slowly, then initially color turning yellow or orange indicate presence of reducing sugar. So this is the comparative table for the testing methods. So ethanol extract, ethyl acetate extract, aqueous extract, and N extract. So that will show the presence of reducing sugars in the plant material. Then next method is qualitative test for carbohydrate. How to identify the carbohydrate from different plant materials. So any large group of organic compounds occurring in foods and living tissues and including sugar, starch, and cellulose, they contain hydrogen and oxygen in the same ratio as water, 2:1, and typically can be broken down to release energy in animal body. Any of various neutral compounds of carbon, hydrogen, oxygen, such as sugar, starches, and cellulose, most of which are formed by green plants and which constitute major class of animal foods. Generally animal food मध्ये याचा जास्तीत उपयोग होतो. ह्याच्यामध्ये पण आपण केमिकल्स दिलेले आहेत. त्यामध्ये इथेनॉल, इथिल एसिटेट, एन हेक्झेन, कॉन्सन्ट्रेटेड सल्फ्युरिक ऍसिड, मॉलिश रिएजंट, अल्फा नॅप्थॉल डिझॉल्विंग इथेनॉल, दॅट इज मॉलिश रिएजंट. नंतर मेथड ऑफ एक्सट्रॅक्शन पण दिलेली आहे आणि हाऊ टू टेस्ट फॉर दिस कार्बोहायड्रेट्स. The presence of carbohydrates was confirmed when 2 ml of extract treated with 1 ml मॉलिश रिएजंट हा मार्केटमध्ये उपलब्ध आहे and few drops of concentrated sulfuric acid which result in the formation of purple or reddish color. So that is the test for the. This is again summary of the test. Ethanol extract, ethyl acetate extract, aqueous extract, and enzyme extract. तर याच्यामध्ये पीपीटी रेड कलरची आली की आपण कार्बोहायड्रेट्स पर्यंत असं म्हणू शकतो. नंतर पुढचं आहे तर आपण बघूया. माझ्या मते हे शेवटचं प्रॅक्टिकल असावं. टू डिटरमाइन व्हेन आयसोलेट नंबर बाय कॅमेरा लुसिडा टेक्निक. याच्यामध्ये आपण कॅमेरा लुसिडा वापरतोय. आपण आधीच्या सिलेबसमध्ये हे बेसिक प्रॅक्टिकल होतं ड्रॉइंगचं. आता व्हेन आयसोलेट म्हणजे काय? When isolate is a small area of green tissue surrounded by the ventlets. The ventlets number is average number of ven isolates per square millimeter of leaf surface. It is determined by counting the number of ven isolates in an area of 4 square millimeter of central part of the leaf between midrib and margin. ह्याच्यामध्ये आपल्याला रिक्वायरमेंट्स काय काय लागणार आहे? मायक्रोस्कोप, फ्रेश लीफ कंक्लूड स्लाइड, ग्राफ पेपर, मायक्रोमीटर, शार्प रेझर ब्लेड, टेस्टिंग स्प्लिट लॅम्प. ह्यात प्रोसिजर काय आहे? तर प्रोसिजर मध्ये आपण जे प्लांट मटेरियल जे आहे ते आपण 1% 30 मिनिटं बॉईल करायचं. अलाउड टू कुक दॅट मटेरियल and then cut a graph paper of appropriate size and make a window of 4 square millimeter on it. Stick the graph paper on the lower surface of slide with the help of gum. Place cleared leaf on the slide superimposing the window. Observe the slide from the 15x eyepiece of the microscope. Trace out the ven isolates from the surface from the window. Count the number of ven isolates from the given area and note down your readings for upper and lower surface. For reference, draw a line of 1 for stage micrometer scale. Take readings for 3 such squares for each surface. Here is the observation table for upper and lower surface. So number of ven isolates in 1 square millimeter, reading 1, 2 and 3 and then mean. And upper surface and lower surface we are taking reading. So conclusion, number of ven isolates is always more on lower surface of leaf as compared to the upper surface. More transpiration. Conclusion. बघा पुढे काय
Thank you, sir.
Yes,
Are there questions?
Due to shortage of time, I could not cover all aspects.
Are there any questions from participants? Sorry, sir. You can present this. One more is there.
One more slide.
Yes.
Triphala Churna.
Yes.
It is also old combination from Ayurveda. It is a combination of 3 fruits. That is बेडा, हिरडा अँड आवळा या तिघांचं हे कॉम्बिनेशन आहे. आणि याचं पण आपण तिघांचं चूर्ण एकत्र करून त्याला त्रिफळा चूर्ण असं म्हणतात. आणि हे आयुर्वेदामध्ये फार वर्षापासून वापरलं जातं. आयुर्वेदामध्ये त्रिफळा आणि त्रिकटू या 2 गोष्टी अत्यंत महत्वाच्या आहेत. त्रिकटू मध्ये सुंठ, मिरी, पिंपळी आणि त्रिफळामध्ये हिरडा, भेडा, आवळा या गोष्टी महत्वाच्या आहेत. त्रिफळा इट इज अवेलेबल इन पावडर, ज्यूस, टिंक्चर, एक्सट्रॅक्ट, कॅप्सूल और टॅबलेट फॉर्म. According to Ayurvedic tradition, Triphala has properties to treat and almost encyclopedic range of health conditions. Some of these attributed to Triphala's laxative, which suggests that cleans the system, digestive system. Depending upon how much is prescribed, Triphala may be used as a bowel tonic at lower doses, elevating gas and promoting digestion, or purgative at higher doses. The chemical constituents of each plant they have been discussed in the next slides of the Triphala Churna. And then every fruit has been described for its external properties as well as internal properties. त्रिफळा चूर्ण करताना मात्र एक गोष्ट लक्षात घ्यायची आहे की त्याच्या फळामधल्या बिया आहेत त्या बिया काढून टाकायच्या आणि उरलेल्या पेरिकार्पची पावडर करायची आणि ती पावडर त्रिफळा चूर्णामध्ये वापरतात. त्रिफळा चूर्ण तयार करताना कसं तयार करायचं याची पण रेसिपी आपण याच्यामध्ये दिलेली आहे. तर या रेसिपी मध्ये आपण बघणार आहोत आणि त्रिफळाचे काय काय हेल्थ बेनिफिट्स आहेत सांगता 12 ते 13 हेल्थ बेनिफिट्स आहेत. विटॅमिन सी त्यामध्ये आहे, फ्लॅव्होनॉइड्स आहेत, पॉलिफिनॉल्स आहेत, टॅनिन्स आहेत, सॅपोनिन्स आहेत. दे हेल्प फाईट ऑक्सिडेटिव्ह स्ट्रेस कॉज्ड बाय फ्री रॅडिकल्स. दे आर व्हेरी गुड अँटीऑक्सिडंट्स. त्यानंतर त्यामध्ये shown protect against certain cancers in number of test tube and animal studies. This herbal remedy also in colon and prostate cancer cell death in test tube studies. Many protect against dental diseases and cavities. Triple has antimicrobial and anti-inflammatory properties that can help prevent plaque formation, a common cause of cavities in gigantivitis. याच सांगता आपण 10 ते 12 युजेस प्रिपेअर दिलेले. तर इथं हे पीपीटी संपते. तर माझ्या मते माझ्याकडे जी प्रॅक्टिकल होती दिलेली त्या सगळ्या प्रॅक्टिकलचा प्रोटोकॉल तुम्हाला ईमेल करतोय आणि तो प्रोटोकॉल तुम्ही सर्वांपर्यंत पोहोचवाल. कोणाचे काही प्रश्न असेल तर त्याबद्दल मी तुमच्या प्रश्नांना नक्की उत्तर देईन. माझा फोन नंबर सरांच्याकडे आहे, तो फोन नंबर घ्या. तर अशा पद्धतीने हे प्रॅक्टिकल चौथ्या मधला बराचसाच भाग हा आयुर्वेदाचा आहे. वास्तविक पाहता मी मग जेव्हा कमिटी होतो इंडस्ट्रियल ऑप्शन मधून तेव्हा मी हे सुचवलेलं होतं. पण तेव्हाच्या ज्या दिवसने बऱ्यापैकी ऐकून घेतलेलं आहे आणि बऱ्यापैकी आयुर्वेदिक प्रॅक्टिस घातलेली आहे. आणि माझा इंडस्ट्रियल एक्सपिरियन्स असल्यामुळं आय मे बी वन ऑफ दी बॉटनिस्ट हू हॅव कॉन्ट्रीब्युटेड समथिंग टू द इंडस्ट्री. हे बॉटनीच्या माणसाला इंडस्ट्रीमध्ये नोकरी मिळत नाही असं म्हणणारे बरेच आहेत. पण मी स्वतः इंडस्ट्रीमध्ये 5 वर्ष काम केलेलं आहे. तिथली लॅबोरेटरी सेटअप केलेली आहे आणि नंतर वारणा नगरला मी आलेलो. त्या दिवशी सरांनी इंट्रोडक्शन मध्ये सांगितलं की जसं मी पीजीला पण शिकवायला होतो विद्यापीठामध्ये आणि नंतर यूजीला पण इथे शिकवतो. तर तुमच्या सगळ्यांचं मला बोलवल्याबद्दल आभार. आणि माझे कलिग डॉक्टर आरडे यांनी मला केमिकल टेस्टसाठी मदत केली, त्यांचं पण आभार मानतो आणि मी इथे थांबवतो माझं भाषण. थँक्यू व्हेरी मच.
Thank you so much, sir. Are there questions from participants? We have requested participants to put their questions in chat box. There is no question in chat box. If anyone wants to ask question, can raise their hand. I think there is no more question. We have Mr. Samson Gaikwad sir, Head Department of Botany, YCIS Satara, as a chairperson. I request him to give his remarks on this presentation. Please, sir.
Hello. Hello, good afternoon.
Good afternoon.
Am I audible?
Yes, you are audible, sir.
Hello. So, good afternoon and welcome to section second. Now, this session deals with applied botany and it is related to 6 different topics of practicals such as biofertilizers, drug plants, adulteration, organoleptic study, phytochemical tests, and Venn isolate number as per camera lucida technique. Now, these experiments, I think this will improve to develop basic skills of the teachers as well as students. I also congratulate Dr. Jadhav sir for his nice presentation and explaining the details of each and every practical. I also congratulate Devadi College, Devadi, the authority of the college, as well as the staff of Botany Department, especially Dr. Vilas, Dr. Anil, and Dr. Suhas, and their teammates for organizing this workshop. I also thank the authorities for giving me a chance to present my views. Thank you. Thank you very much.
Thank you, sir. To this session, Dr. S.Y. Jadhav sir, Head Department of Botany, Yashwantra Varna College, Varna Nagar.
You are the age of old age also.
Presented well-defined presentation and precisely and easily explained all the practicals. For that purpose, I am very much thankful to you. I also would like to convey my heartfelt thanks to Mr. Samson Gaikwad sir. With this session is over, and immediately we will go for session second. I am going to hand over this session to Miss A.B. Mamleya madam. Please. Mamleya madam.
सुहास सर.
Yes sir.
त्यांना अनम्यूट करा.
हा केले sir.
Okay, sir. Thank you. Thank you so much, sir. Good afternoon, everyone. Myself, Miss A.B. Mamlaya, Assistant Professor, Department of Botany, Dahibadi College, Dahibadi, and welcome back to second session on revised syllabus of B.Sc.III practical based on paper number 12 and 13. Let me introduce our first resource person of second session is Dr. S.M. Moholkar sir, Assistant Professor, Department of Botany, RS B.M. Aund. He has completed his PhD on the topic entitled Diversity of some. He also bagged one minor project during his journey in the same profession. About his research, sir has published number of research papers in reputed journals. In addition to these, he has conducted seminar on impact of vehicle air pollution on environment. Sir has attended more than 30 seminars, workshops, conferences during his career. He presented more than 11 papers in national and international conferences. He also acted on various bodies like working as university practical and theory examiner at B.Sc. first, second, and third. Nonetheless, also serving as ADAP BIOS chairman at Horticulture, Science and the Technology. Sir is also working as exam coordinator. He had experience of serving as NSS member in year 2004 to 2006. Except to this, Sir also contributed in social work by donating funds or by actively participating cultural activities organized by various colleges and universities. Now, I would like to request Dr. Moholkar sir, with the permission of our chairperson, Dr. Rekha ma'am, to share his knowledge with us regarding revised syllabus practical 4 of based on paper number 12 and 13. Please, sir.
What, sir? What, sir?
Yes, sir. अनम्यूट केले सर त्यांना.
मोहोळकर सरांना को होस्ट केलंय का पहा.
ओके बघ.
स्क्रीन शेअरिंग ऑन. कॉम्प्युटर ऑडिओ म्हणजे तिथं डायरेक्ट चालू होईल आता लगेच
मॅडम
सर को होस्ट केले त्यांना.
हा त्यांना स्क्रीन शेअरचा ऑप्शन द्या.
Good afternoon to everybody. Thank you, ma'am, to introduce me. I would like to express the thanks of Dayodi College, Dayodi Principal Dr. Balwant sir, Head Department of Botany, B.B. Kamble sir, Dr. Anil Jagtap sir, and their colleagues. The practical course, which is based on paper number 12 and 13, which is allotted me to share some views with you. These are the 11 practicals. All you are familiar with these practicals, that is the studying of budding techniques. In a budding techniques, there are two types. One, that is the patch budding, and second one, that is the tree budding. Second practical in which the layering technique. Under the layering technique, there is a one technique enrolled in syllabus, that is the air layering. The third practical, particularly for grafting, and in the grafting, there are two types. One that is the whip, and second one that is the approach. The fourth practical, that is the potting and repotting. All of you are familiar with that one. Fifth one, that is the demonstration of bonsai. Sixth one, that is the bottle garden and hanging baskets. Seventh one, that is the flower pot, flower bucket, and flower rangoli. Eighth one, you are also familiar with that one, that is the garden implements. Ninth one, that is the ornamental plants. There are 3 plants that are enrolled in our syllabus. The 10th practical, that is the study of edges and edges. And the 11th practical, that is the study of indoor plant. First of all, the students know the definition of budding, and then we'll switch over the patch budding. To solve the difference between that two ones, the requirements required for carrying out the patch budding, that is the sharp knife, polythene strip, as well as the plant materials, those are utilized, particularly the mango as well as the walnut. These are the steps. We will not discuss detail about that one. I will share my presentation to Dr. Anil Jagtap sir to circulate all participants. In that, all these steps, the role of a polythene strip, it is a very important one, and that role you told the students. Similarly, another practical, that is the budding. That budding, first of all, you told the definition of that budding. Then students know what is stalk, what is scion, and for carrying out that practical, you told some equipments, those are required for carrying out that practical, that is the budding knife, polythene strip, and the plant material particularly utilized, that is the citrus as well as the rose plant. The steps are up to you, you are familiar with that steps. After that one, another practical, that is the study of layering, and in that practical, that method is the propagating new trees and the shrubs from the stems still attached to the parental plant. First of all, you compare that practical with budding as well as the layering. What is the difference in between that two ones? Because all these methods, these are the asexually propagated plant methods, and students know what is the difference in that two ones. For carrying out the practical, the sphagnum moss, which is the essential for development of the root tips. The requirements, that is the sphagnum moss, sawdust, vermicompost, polythene strip, knife, as well as the thread, which is necessary. In which plants, particularly that technique which is carried out, particularly in protons, ficus, guajava, and pomegranate. These steps, you are familiar, all these ones. Then another part, that is the whip grafting, or it is also called as a slice grafting. Particularly, that method, that is the surface of scion and rootstocks are brought together by their respective ends and joined by overlapping and binding together. That method, which is the oldest method, it is also a asexual method. By using this method, you can propagate such type of the plants. Mostly that method which is applied particularly in pear as well as the grapes. Student know the what type of the plants as well as the other materials those are required one, that is the sharp knife, polythene strip, particularly that method which is run in a pear as well as the grape. These are the steps. First of all, you can select the 1 year old material having 8 to 15 mm diameter. The cyan and the rootstock should be an equal diameter. This is the very important one while you can carrying out that practical, and all the sides of branches these are removed from the both. Cut the rootstocks, plant with a seccator at a 20 to 25 centimeter from the soil surface by using the sharp garden knife, a long sharp sloping upward cut which is given on the rootstock. Then select the cyan plant, stem portion having the same diameter, which is a very important one while you carrying out the practical. A long slanting cut which is given on that cyan plant on the lower end by using the sharp knife. Cut end of both ones, that is the cyan and rootstock. Students, first of all, you can clear the definition of that cyan as well as the definition of that rootstock, or what is the difference between cyan. You can told clearly. Then and then these practicals effectively understand by all the students. Then these are the steps. Sequentially, you can carry out that another practical which is related with the sexual methods. Then you are also familiar with that practical, that is the potting and repotting. We will not discuss details about that one. These are the steps. Overall, there are about 7 to 8 steps in which the potting as well as the repotting which is carried out. I focus on that practicals. I think these are the new one, that is the demonstration of bonsai. Student know the definition of bonsai. That means the tree grown in a container or a tree planted in a dish or pot. It is called as bonsai. That technique which is coming from the Japanese, and you told that part in a theory, but the students know the technique which is coming from which a nation. Another definition is also given, that is the miniaturized tree grown in a dish, and it resembles in all respect its a large counterparts in a nature. Then in a demonstration of bonsai, the another part which is very important, that is the selection of plants. Such a plant must be 2 years old. Tall growing plants must be select. That plant should be a suit to the atmosphere in which it grows. A small leafy parts are selected. The plant must be a evergreen. These are the criteria for the selection of plant. After selection of plant, the selection of container, which is also very important because it should be harmoniously matched with our plant. What type of a style which is given to our plant, and on that one you can select such type of the containers. There are various types of the containers. They may be earthen pots or ceramics or plastic pots. These are also used for plantation of such a bonsai plant. These are the equipments those are required for carrying out the practical. That is the seccator, small saw, so on. The containers of the pots as well as the other material which is also required, that is the garden soil as well as the compost as well as the pieces of bricks and sand. These are the 3 kinds of the mixture you can utilize it for preparation of such a bonsai. That is the bottom layer. Students know the bottom layer, what is the mixture of that bottom layer, then middle layer, what is the mixture of that middle layer, and then upper layer, what is the mixture of that upper layer, or the composition of all these 3 layers. While in a exam, the demonstration is carried out. In an oral, just you ask what are the 3 layers and what is the composition of that 3 layers. After mixture, then actual method of potting. Keep a piece of nylon net over the drain holes of that container. Insert a copper wire. You should explain the role of that copper wire. Why the copper wire which is utilized instead of metal wire. The scientific reasons are given to the students. What are the benefits of that copper wire as well as the other things? What is the role of nylon net? Then place a plant in a pot and tie with a copper wire firmly. The main shoot and the few roots, these are usually tied properly. The poor bottom layer mixture at the base. Above that, keep a second layer, that is the middle layer. Finally, add a upper layer mixture, keeping a small distance at the top. Watering is done by keeping this pot in a big container containing a water or with spare. That means after plantation of that plant in a container, you should not directly provide the water. Just you can dip such a pot in a large container with water. Such a potted or repotted plant should be kept in a shade. This is the scientific reason. Do not keep such a plant directly in the sunlight. Such things you told the students because the harm is seen in such a plants. Then a suitable plants those are utilized for carrying out the bonsai, that is the Adinsonia digitata, which is the best plant for preparation of bonsai. Then another which is easily available in our area, that is the Ficus bengalensis. Then Milia azadirect. In another one, that is the Phyllanthus empirica, the best bonsai which is also prepared by using the Bougainvillea spectabilis. Sometimes the Morus alba or Picomus transplants, these are also utilized for preparation of bonsais, out of which any plant you can select for carrying out that practical. Then another concept in that practical, that is the bottle garden. First of all, you told the definition of that bottle garden to the students. That means the bottle garden is the container similar to the terrarium in which the plants they are grown. They usually consist of the plastic or glass bottles with a narrow neck and small opening. Such plants they are grown inside the bottle with a little or no exposure to outside the environment, which requirements for carrying out the practical, that means the glass or plastic bottle, long handle spoon, cutter, pebbles, water, so on. What are the steps while you can carrying out that practical? First of all, you can select the bottle, then clean it properly, dry it, and then fill the bottom of bottle with pebbles. The students know the role of the pebbles. You should told what is the role of the pebbles. That means they maintain the aeration. Then third step, add the thin layer of activated charcoal as well as the peat moss. Should also told the role of that two ones. And then you can plant your gardens. Such a garden, for preparation of bottle garden, generally small growing plants mostly utilized. A small size of plants, they give a better effect. Then after a selection of plants, then potting up your plants, the last stage, that is the seal of your garden and watch it for growing. These are the steps where you can carry out that practical. You can run that practical by through that all these steps. What are the uses of that bottle garden? Students know why can prepare such a bottle garden. What are the uses? No doubt which is the theoretical part, but orally you can ask or you can assist the student. What is the purpose of preparation of bottle garden? Student know the uses of that bottle garden. Then another concept in that practical, that is the hanging basket. First of all, you told about the hanging basket. That means a container made up of wood or wire to resemble a basket to hold a plant hung up for a decoration or for a greenhouse cultivation as a orchids. Or another way we can say that is the basket or a similar container which can be suspended from a building by a small rope or a chain, and in which a decorative flowering plants are grown. Such baskets they are hung to the ropes or terraces or in a verandas or in a drawing room or under the shade of trees. That is the place where you can mount or where you can keep it. These are the steps while you can prepare the bottle hanging basket. While you can carrying out the practical, students know there are different types of the hanging baskets. The most popular, these are the made up of galvanized wire, and they are very light, sturdy, and last for the years. Sometimes the plastic baskets are also used for preparation of such hanging baskets, or sometimes a teak wood or cypress wood baskets are also used or preferred for the preparation of such a hanging basket. Sometimes the ceramic pots or terracotta pots are also used for planting such a hanging plants. For preparation of such hanging basket, these are the requirements, like a basket usually lined by a thick layer up to 2 inches of porous sphagnum moss. Wet the sphagnum moss before pressing it to the bottom and side of the basket. Then coconut fibers are also required, and landscape fabric, which is also required one. Sometimes some practitioners, they also use a plastic film or an aluminum foil instead of moss. When these materials are used to line the basket, punch the small holes in the bottom of the lining material for draining and aeration. That means you told the roll of each and every material which is utilized for the preparation of hanging basket. That means the roll of plastic or aluminum foil. Why you can punch that foil. This is the steps for the preparation of hanging basket. That is the take the hanging basket, line the white punched aluminum foil or a plastic paper, then partly filled with a coconut fiber of moss. Then plant raised either by seed or cutting. Plant may be erect with a height or pendant with drooping branches and colorful leaves. The remaining part of the basket, which is filled with 2 parts of loamy soil. Students know what is the types of soils, and you should give a definition of that loamy soil, as well as you can add the 1 part of a dried cow dung. The watering of such a hanging basket hung down from the roof, and any crossed arm using a nylon thread. The care should be taken while you can prepare such type of a hanging basket. That hanging basket, which is watered regularly, and after some intervals, you can add fertilizers, that is the NPK fertilizers, or in the market the liquid manures is also available. Such requirements for blooming as well as growing such plants, you can give like intervals after intervals. Which plants those are suitable for hanging basket? That is the adiantum, then begonia recommends, impatiens repens, Tradescantia, as well as the vanda or verbena. These plants, these are suitable for carrying out that experiment. Then another sub-practical in this one, that is the preparation of flower pots. And for the preparation, the vases are also important. That means the selection of the vases, as well as the selection of the container, size and the shape of the container, which is also depends upon the type of the flowers. Those are arranged, meant to be a display. That means the various types of the containers these are made up of. Students know the different types of the containers. That is the by which material which is utilized for the preparation. That is the porcelain or ceramic, brass metals or bamboo flower pots are also used, or containers are also used. That containers should not be a bright and made up of glass, because they attract the beauty of that flowers. For completion, the selection of flowers, which is also very important. That means the various or the same color the flowers with foliage or a fresh or dry twigs are also used for the preparation of such a flower pots. The conditioning of the flowers, and making the flowers to remain fresh for a long time, it is called as conditioning of flowers. Students know the definition of that conditioning of flower, because it increases the vase life. That means the plants which depends upon the temperature as well as the relative humidity, holding water capacity, light and air. The following methods these are also useful to extend their vase life. The flower should be cut at the half open stage or bud stage. The flower should be cut along with a stem of at least 5 centimeters length and 1 or 2 leaves. The stems of the flowers should be cut under the water and kept in a cold water until they are arranged. That means these are the steps you can carry out while you can prepare such flower pots. Some flowers they remain fresh for a long time if they are in water or in a vessel. Mostly the lily or iris or caladium, generally they are remains fresh. Sometimes in some flowers, the buds are also important one. The opening of the buds should be wrapped with a transparent tape to delay the opening, particularly in a rose plant, or beside this, the cut the end should be covered with candle wax or egg white. Students know the role of that wax as well as the role of that egg white or the tape to delay the opening of the flower. This is the main role of that material which is utilized for wrapping such a buds. The lower leaves of the cut twigs should be removed and the twigs should be kept in a cool dark place until they are used. This is the tricks where you can prepare the flower pots. Sometimes in a lily, the stamens should be removed because they created means removed because they coated with wax or to prevent the pollination. Because while keeping such a flower pots in a house, sometimes some pollens they are allergic one, and you can take the precaution to remove such type of the anthers from the flower. This treatment extends the vessel life of lilies up to 10 days. This is the role of the materials mostly used. Another part, while you can prepare the pots, the arrangement of the flowers in a container, a suitable vase or saucer particularly is taken, which is filled with water containing a sucrose 2% and ammonium sulfate, that is the 0.4%. Students know the combination of that two ones, and this water extends the vase life of the flower. What is the role of that sucrose as well as what is the role of that ammonium sulfate? They delay the ripening of the flower, and this is the role told the students. For a one day preparation of such a flower, above combination which is not used in this container, such a conditioned flowers are then arranged beautifully in a container. The flowers, foliage, twigs, and fruits should be arranged in such a way that to bring out the beautiful color combinations. When you arrange such type of the flowers, contrasting colors you can choose as well as the colored leaf you can also choose. When you can select the flowers, all such stocks of the cut flowers must be originate from a single point. That means the base of that cut flowers, which is originated from the single point, because it gives a beautiness of that flower pot. Expose such stock of the cut flowers should be 1.5 times the height of the container. That means that ratio, proportional ratio you can maintain, then and then it gives the beauty. Then these are the different steps to develop such type of the flower pots. Various types of the flower pots you can utilize for the preparation of such a flower pots. Then a new practical which is added one, that is the buckets. First of all, you told the definition of that bucket. It means a bunch of cut flowers for carrying out in a hand. It is called as a flower bucket. It represents the gift or the expression of the praise or the compliments. The simple bucket enhance and elaborate the beauty of the plain dresses. Then a flower bucket is the collection of flowers in a creative arrangement is seen. The flower buckets can be by the Victorian era or almost every flower had a specific meaning. While you can prepare the bucket, the color of the flower or the flowers those are utilized, they are having some specific meaning. Such type of the flowers you can utilize for the preparation of such a buckets. Then choose your flowers color freely. Traditionally, mostly the white flowers are generally utilized for the preparation of such a flower buckets. But recent trends to hold the flowers of all the colors, those are utilized. For the preparation of such flower buckets, mostly the red roses or calla lilies or poinsettias are also popular for the preparation of such buckets. Such contrasting colors are also another popular choice. For example, the pink flowers look beautiful with the blue flowers. That means such type of the selection is depends upon the who is prepared such a bucket, and what are the ideas in their minds on which such type of the flower buckets are also prepared one. Sometimes the flowers that allow the traditional buckets in a single or a varieties of shades. These are the color combinations of the ribbons. Those are also utilized for tying such a buckets. A long flowers look beautiful cascading down from the front end of the flower buckets. Always purchase more flowers than you think will need, because at the time of preparation, then you can think on that one. The ideas are also enhanced one, and that's why for fulfilling your need, you can purchase a large amount of flowers from the market. Such a fresh flowers, those are delivered and in hand, may inspire a new ideas or the additional of buckets. First time the bucket makers may make the mistake or change their minds. These are the steps where you can prepare the buckets. That means the removal of the coating of that flowers from their package. Then second one, that is the gather 4 or 5 blossoms of the flower for making such a flower bucket. Then finish the bucket with the outer ring, which is also very important one while you can prepare such a bucket. Then wrap the flower tips around the stem to hold all such a 2 or 5 things. Then another step, that wrap the ribbon around the flower. I think you observe that when you purchase the bucket from the market, at the base, such type of the ribbons, these are also wrapped one to enhance the beauty of the flower. Then sixth step, that is the spray the flower heads with a hairspray. That spray with a hairspray, let dry for a few minutes. Fill a vessel with enough water to cover the stem ends, but not the ribbon just above it. This precaution you can take, and you can place the buckets in a vessel. Then another concept which is related with practical number 7, then the flower rangoli. It is an art of making a beautiful flower carpet using attractive flowers and leaves. It is usually made during the Diwali or in a Tihar, or particularly in a Sankranti and other Hindu festivals in India. Students know the concept of the flower rangoli, from which tradition it is coming out. And particularly in Hindu festivals, such type of the rangolis, they made in front of their houses, or in any occasion, they prepare such type of the rangolis. That means that designs, these are passed from one generation to the next, keeping both the arts form and the tradition alive. This is the role. That's why that practical, which is enrolled in that syllabus, you can, the students know the tradition, and our goal is to maintain our tradition. The purpose of the rangoli is fill the strength, generosity, and is up to bring a good luck. It is traditionally done by mostly the girls as well as the women, although the males and boys, they are also prepared one. That rangoli design can be a simple as well as the geometric shapes, deity impressions, but they can also be very elaborate designs which are crafted by numerous people. The best material is usually the flower parts, maybe a dry one or maybe a wet one, for enhancing the beauty as well as the freshness. Sometimes a sawdust which is also utilized to spread uniformly over the carpet, and then sketch the diagram or a design, and then you can put such type of the different flowers for the preparation of such a flower rangoli. Over time, the imagination and innovative ideas in the rangoli, that art have also been incorporated. The hospitality as well as the tourism industries, in which such type of the traditional things you can give to the newly coming students. Then these are the different steps for the preparation of rangoli. All these photographs, my students in the last year, they prepare such type of the rangolis. I organized one competition, and just to give idea how to design by utilizing the flower parts as well as the leaves to develop such type of the floral rangoli. The purpose is also known to students, and the purpose of that rangoli is to feel the strength as well as the generosity and not bring the good lucks. It is traditionally done by the girls as well as the boys. Another practical which is also traditional one, that is the garden implements. The student know the implements, their construction, their use, and what should, why you can use such a implement. After implementation or utilization of that any implement, the care is essential. What type of the care you should take after utilizing that instrument, you should told the students, because it increases the efficiency of such a garden implements. These are the 5 you just I select. Then another part, there are 3 plants. Those are enrolled in our syllabus. That is the first one is the rose. That rose which is easily available in our area. Students know that their botanical name, family, their center of origin, and just give the gross morphological characteristics or unique characteristics. Those are related with that flower, as well as the students know the ornamental value of that flower. Mostly that rose which is used for decorating the garden plants as well as the cut flowers, or the many species are grown in domestic gardens. Similarly, the another plant mostly in the market which is utilized, that is the gerbera. And first of all, you told the name of that plant, botanical name, their family, as well as the common name, and the center of origin, and some gross morphological characters, which is very important one. While the students, they go to the market and while observe the flowers, but the students, they know the name of the flower, and that's why you can create the awareness among the students. Such type of the characteristics you can explain a very simple manner, and then told the role of that plant, particularly in a ornamental. That plant which is also used as a decorating the garden plant or as a cut flower. Many species these are grown in a domestic gardens. Another third plant which is also used, that is the edges, that you told the family as well as the center of common name, the common name of that plant and the flowering period. Just told the in short their gross morphology and the uses or the ornamental value of that plant. Then another part is also enrolled, that is the hedges and edges. Student first of all told what is the difference between the two ones. That is the definition of hedges as well as the definition of edges. What are the unique characteristics of hedges and edges? What is the exact difference between the two ones? And once the students they know the difference between the two ones, then they remember that plant which is used for as a border plant, and that's why it is called as hedge plant. I can select only 2 or 3 plants. Those are easily available in the garden. First of all, students know the botanical name of that plant, their family, their center of origin, and a brief gross morphological characters of the plant. Then another plant, that is the Hemilia patens. Same pattern is also used for describing such plants. Then another one, that is the Phyllanthus myritifolius. When you can visit a Koina garden, and in that garden, such type of the fencing which is utilized, and students they ask, what is the name of that plant? While you can give a live performance or live things to that plants, they remember forever that plant. That is the name of that plant. That is the Phyllanthus, and that plant which is observed in a Koina garden. That means that plant which is also used as a hedge plant. And just give an idea about that plant, that is their gross morphology as well as the other characteristics. Then another plant which is utilized as a hedge plant, that is the Lawsonia inermis. In Marathi we can call that is the mendi or henna, particularly you can call, particularly in Konkan, that plant it is utilized as a hedge plant. Students know the gross morphological characters of all these plants and their ornamental value. You told the characteristic of that plants. Another plant, that plant belongs to Verbinaceae, and same characters you told the students, that Botamaria inervis, It is the botanical name, and flowering period particularly in the plant is November to January. That is the another concept which is related with that one, that is the hedges. And students know what is the difference between that two one, that is the hedge and hedge plant. I can select only two plants which is easily available in all colleges or campus. That is the Alternanthera amabilis. That plant belongs to family Amaranthaceae. Their common name, that is the calico plant. You should told some gross morphological character or a distinguishing character on which these plants they are easily identified by the students. That is, these practicals they create the awareness among the students while they observe the plants in a field. They definitely know that plant, that is the Alternanthera amabilis. Then another plant which is also used, that is the Justicia genderosa. The family is Acanthaceae. Their center of origin, that is the India as well as the Southeast Asia. Same gross morphological characters you told the students, and on that basis they can identify the plants. Then another practical, which is a enroll, that is the study of indoor plants. The student must know the outdoor plants and indoor plants. What is the difference between the two ones? Some unique features of that indoor plants, first of all, you can told to the students before carrying out the practical. And then there are few plants you can select as a indoor plants. Mostly the plants those are used in a house plants or as a grown in a house, generally it is called as indoor plants. And after telling that definition, the same pattern which is also used to told the characteristics of that students and what is the ornamental value of that plant. That another plant, that is the Asparagus primosus. The family, it belongs to Asparagaceae. The ornamental value is also very important. That means the leaf-like cladophyles are also having the ornamental value, and you can simply told that's why that plant which is categorized under the ornamental, and that plant where it can grown in a house, there is no any harm. That plant they are well flourished. That means that plant which is the fast growing, as you told some unique characteristics related with that one, and which precautions you can take when you can plant the plant in a house. That means it requires a rich as well as the slightly acidic, well drained soil. That means these 4 or 5 characters are essential while you talking about such a plants. Then another plant which is also utilized as an indoor plant, that is the Diphendicia amina. That plant belongs to family RSC. The ornamental value or ornamental part, that is the leaf part, because that leaves, these are the variegated colored. This is the unique feature for identifying that plant from the field. Control some characteristics, those are related on that one, as well as the conditions those are required. Because for growing that plant, it requires high humidity and a draft-free atmosphere. It should not be placed in the front of an air conditioner unit or fan. While you can watering that plant, that leaves becomes straight as well as the brittle, and their beauty which is a lost one when you can keep such a plant in front of a fan as well as the air conditioner. Another plant which is also used is the cypress. You can told the ornamental value of that plant, that is the leaf part. Whether it is annual or perennial, you can told that characteristic, that is the presence of rhizomes, and the plant it requires more than 2 years for the completion of the life cycle. That terminologies you can clear one while you can told some characteristics related with that plant. Then you can told the same characteristic, influences of that characteristic, and the conditions those are required one for growing such a plants in our house. Then another plant, there is the Spathiphyllum, and that Spathiphyllum, that plant also belongs to Araceae, and the ornamental plant, particularly the leaf part and the inflorescence, which is a very attractive one. And students know the inflorescence type, what type of the inflorescence which is present, because that's the ornamental value. All such plants, they are evergreen perennial herbs. The leaf size, these are the broad one, dark green color. What is the difference between Diphendicia and Spathiphyllum? You can compare their two plants. easily students they clarify these two plants while you can compare these two ones. Then what precautions you can take? Because it does not require a large amount of water and light to survive. That watering is approximately once a week. It means to completion of 6 or 7 days, you can water it one. It cleans the certain gaseous environmental contaminants such as benzene as well as the formaldehyde. While such gases those are available in our houses, they are easily removed, and that's why such a plant they are keep it in a house. Thank you for listening me. The one slogan that is the slave nature for future. That presentation, which is available on my departmental website, I share that website, and in an e-content, that material which is available one for preparation of such a presentation. These references mostly utilized that horticulture by Kumar Sen, which is published by Saras Publication, and handbooks of horticulture, which is published ICAR New Delhi. Thank you for listening me. Thank you once again for given opportunity for share my views with us. Thank you.
Okay, thank you so much, sir, for sharing your extensive practical knowledge and guide us. Now, chairperson for this session is Dr. Rekha Devkar Madam, BOS member and Head Department of Botany, Deshbhakt A.B. Naik Arts and Science College, Chikhali. I would like to welcome on behalf of Department of Botany, Dahiwadi College, Dahiwadi. I would request to our chairperson, Dr. Rekha ma'am, to share her suggestions and opinion regarding this. Rekha ma'am, please.
Hello. I am
Yes, ma'am,
audible.
please. Yes, ma'am.
Okay.
Yes.
Good afternoon. Okay. Good afternoon. I'm very grateful to chair this session with all of you today. First, I want to congratulate all organization team for organizing such nice workshop. Then today's resource person for this session is Moholkar sir, gives very nice, knowledgeable and energetic presentation. He explained all nursery techniques in very short but detailed information. He completed all topics assigned to him, such as budding, air layering, grafting, potting, repotting, bonsai preparation, then bottle garden, hanging basket, preparation of flower pots, bouquets, floral rangoli, preparation of flower pots, floral rangoli, garden implements, study of ornamental plants such as roses, gerbera, hedges, edges, and indoor plants. He prepared his presentation very nicely with nice photographs. Then he covered all, I think all maximum point of the syllabus of this paper, practical. Then he present his slideshow with bottom-up heart and very energetically. Then lastly, I am very thankful to the organizing team of this workshop of Dahiwadi College, Dahiwadi. Especially, I want to thank Principal Dr. Balwant sir and Dr. Anil Jagtap sir. I feel very proud today because I am the ex-student of the Dahiwadi College, and this workshop is very nicely organized. So I here declare the today's third session is completed. Thank you very much.
Hello.
Hello.
अनिता मॅडम
हाँ बोला सर.
बाबू सोनार सरांना इंट्रोड्यूस.
Okay, sir. So thank you so much, ma'am. Now I would like to introduce our second resource person, Dr. B.A. Sonar sir. Reveal brief biodata of Dr. Sonar sir. He has completed his PhD under Shivaji University, Kolhapur. He is serving as assistant professor of Department of Botany, College B.Sc.III. He has 10 years teaching experience at colleges and universities. Now, about his research experience, he has published 10 research papers in UGC Care journals. He has attended more than 40 national, international conferences, seminars, and workshops, and presented more than 30 research papers in national and international conferences. Sir also has working on college level minor research project. In addition to this, he has worked as resource person, examiner, and evaluator for various workshops, conferences, and competitions. Sir also served as a coordinator of Campus Development Committee and a member of RUSA, B.Sc.III and BHOP program. I would like to request to Dr. Sonar sir to share his knowledge with us regarding revised syllabus, practical number 4, paper number 12 and 13. Sonar sir. Hello.
Sonar sir. Sonar sir.
हॅलो हा आवाज येत नाही तुमचा माझा येतोय का तिकडे आवाज थोडा जरा होय का बरं ठीक आहे
नाळे मॅडम त्यांना स्क्रीन शेअरचं ऑप्शन द्या.
हो सर को होस्ट केले त्यांना ते शेअर करू शकता.
गुड आफ्टरनून टू ऑल ऑफ यू. This workshop is organized by our sister concern, Daivadi College, Daivadi, and their IQSC NAC with under the Shivaji University, Kolhapur. Online workshop is on the revised syllabus of B.Sc.III Botany for paper number 3 and 4. Department of Botany has taken initiative and organized such workshop for the revised syllabus. Myself, Dr. B.A. Sonar, Assistant Professor, Department of Botany, Balwant College, Beta. I am here to present my presentation on B.Sc.III Botany syllabus, practical number 4. We'll see here under the practical 4, we have almost 20 practicals, but I have to present from 12 to 20. In that, first is a qualitative test for sugars in plant material, and second test for starch cellulose, third for proteins, lipids, and 16 is sugars by ascending paper chromatography, determination of fatty acids, and thin layer chromatography, determination of isoelectric point from plant proteins, and visit to nursery. These are my practicals to discuss with you. Very first practical is of qualitative test for sugar from plant material. All of you are knowing that the sugars, they are of two types: reducing sugar and non-reducing sugars. For to perform qualitative tests for in plant materials, we have to, there is principle that carbohydrates are the compounds which are distributed all over in the living plants or organisms. Carbohydrates, they are synthesized by the plants with the help of process of photosynthesis, that all of you are knowing that. And after the process of photosynthesis, we have the molecules like carbohydrates, and these are when dissociated, they show presence of monosaccharides, oligosaccharides, and polysaccharides. And from that, monosaccharides, oligosaccharides, sugars, there are some these are the sugars, and polysaccharides, these are the non-sugars. Qualitative test for sugar, we have to reducing sugars when heated with alkaline copper tartarate. We got the form of copper to cupri, and thus a copper oxide filling solution gives us a color or formazon called as red color formazon with filling A and B solution. There is a well-known test for it, and in the procedure we see that when we take a material, onion leaf material extract, plus if you add equal amount of fillings A and B solution, and boil and wait for 10 minutes, you'll get a brown color PPT. So that PPT confirms us that the onion leaf extract is having reducing sugars. So this is the practical for reducing sugars. And in one more hand, we have another practical, that is qualitative test for sugars in plants for non-reducing sugars, that means polysaccharides. That the non-reducing sugars, they are when hydrolyzed with concentrated sulfuric acid, presence of sodium bicarbonate gives give reduced form of sugars. That means macromolecules, they are reduced to the smaller molecules or monomers of the sugars, and they ultimately react with the filling solution. The same assay was there to show the non-reducing sugars also. There is a test tube set of 2 to 3 test tubes. You have to take the filling solution. Filling test is there, and onion leaf extract we have taken test tubes, and with the help of test tube holder, we can boil the material for the 10 minutes along with the filling A and B solution, and cool and filter. And then after cooling, the concentrated H2SO4 and NaCO3 filling solution and be added and boiled for the 10 minutes. You'll get these 2 test tubes. First test tube gives no red color PPT, and in second test tube you'll get brown color PPT. That means the 2 test tube sets, when they itself confirm that in first test tube you have the polysaccharides like non-reducing sugars, and When we add H2SO4 and NaCO3 to it, it reduces to monomers, and the test for non-reducing sugar is confirmed. So this is all about the first practical, that is reducing sugar. Then qualitative test for starch. This starch is one more such material, and cellulose, which is as a result of photosynthesis that material is formed. Starch is an important polysaccharide. It is stored or storage form carbohydrates. You may have the sources of starch as root tubers, stem, fruits, etc. Starch is composed of two components, namely amylose and amylopectins. There are starch when we dissociate or when it is degraded, will have two types of molecules, amylose and amylopectins, or chemicals you may say. For that, requirement is of I2KI solution, concentrated concentrated NaOH solution. That this is age-old practice for starch estimation. For qualitative test of starch. Then in potato tubers, sweet potato you may choose for the as a material. For that purpose, you have to cut the potato surface and apply I2KI solution and observe. When we take it test for starch, the thin sections of potato tubers, they show the blue-black color in first test tube. You'll see cut surface of the potato tubers plus smear of I2KI, and that gives us blue color in the that potato tuber, cut piece of the potato tuber. Then presence of starch is confirmed. Then in cut thin sections of the potato tuber, again smear with I2KI and observe under microscope. Shining starch grains which are of blue colored under microscope you may observe. This confirms the starch in the that cut thin section of the potato tuber. Then the test for the cellulose. Cellulose is also a macromolecule and major structural component. That is one more polysaccharide. It is almost abundant in complex organic forms in nature. It is composed of glucose units joined together and repeating units of the polysaccharide. Cellulose shows a numerous cross-linking of plant cell wall and made up of cellulose. Then the main source of cellulose is wood, cotton, and other fiber material. For that purpose, chemicals used are I2KI solution, zinc chloride, iodine, etc. The watch glass is needed for this practical. White saucer, glass slides, etc. Material may be chosen as cotton wool, strip of the Whatman paper. You take pieces of the material that may be, you have chosen, and watch glass. Take in watch glass and add few drops of to it. Take some plant material in watch glass and add I2KI solution. Then some of the plant material in watch. Add watch glass. Add few drops of concentrated H2SO4. So these three tests can confirm if there is no change at all, the cellulose is absent. Then if there is no blue color, then cellulose never shows color with the I2KI. Then in plant material plus I2KI, material slowly dissolve and shows solution with the H2SO4. I think this is the assay for the plant material, proteins in the plant material. Then the plant material perform when proteins are the macromolecules composed of one or more polypeptide having in them, and the sequence of the polypeptide amino acids confirms the protein. or nature of the protein. Protein is nothing but a polymer of amino acids. Proteins perform variety of the specialized and essential functions in the plant cells as well as animal cells. The proteins are primarily responsible for the structure and strain of the body. The proteins dynamically functions as enzymes, hormones, antibodies, storage proteins, etc. Then it constitutes about 12% of the protoplasm. Then proteins mainly supply nutrition. Then the plant source are nut. For the proteins, there are plant sources as like the nuts, pulses, cereals, etc. Then the requirement for this these chemicals required for this practical are 5% NaOH, 1% CaCO4, 1% neem hydrine. Then test tube holders, water bath. Germinating seeds of Phaseolus are the plant material for this. Take 20 to 30 gram of plant material, crush it into 20 to 30 ml of distilled water and filter through muslin cloth. And such filtered material is used as a test solution. Then we have to perform two tests, biorate test and neem hydrine test. For that, a qualitative test of protein. Then in the biorate test, we have to take 3 ml of test solution in a test tube, 2 ml of 5% NaOH, 2 drops of 1% CuSO4, mix it and heat, and a violet color is generated. And then in alkaline medium, the peptide linkage in protein molecules react with the CuSO4 to form violet color complex. So that confirms the presence of the proteins in the in that test solution. Then neem hydrine test, all of you are knowing that amino acids are giving blue color with the neem hydrine. 2 ml of test solution plus 5 drops of 1% freshly prepared neem hydrine solution is taken. That gives us heat it, and that gives us blue color. That violet color complex is showing presence of proteins. Then violet color confirms the presence of proteins in the test solution. This is qualitative test for lipids. Lipids are the molecules. These are also macromolecules. These are present in the naturally. They are lipids are the group which are naturally occurring substances which are insoluble in water but soluble in organic solvents like ether, chloroform, alcohol, etc. And chemically, lipids are defined as esters of fatty acids that we know as the fatty acids, or as the triglycerides of fatty acids are building blocks of the lipids. Fatty acids are called as building blocks of the lipids. The lipids are the important constituents of the diet due to their higher energy value. The phospholipids and glycolipids are the structural components of the membrane. The lipids yield alcohol fatty acids on hydrolysis. Lipids widely distributed in the plants. In plants, they occur in the seeds and fruits. For this practical, we need chloroform, 5% sodium bicarbonate. For this purpose, we also need glasswares like test tube, a test tube holder. For this, there is no need of water bath or soap. Plant material like vegetable oil, any vegetable oil you may choose, groundnut oil, olive oil, etc., or castor oil, or sometimes you may mustard oil. Take 2 drops of the oil in test tube, and in the sample test tube, 2 ml of the distilled water, and shake well to observe. In the sample, you may find in the test tube the sample remains undissolved. That test tube containing water plus oil, that is oil is not miscible with the water, so that is there as it is, so it is called as the inference is lipids present. And one more solubility test is there. Take 2 drops of the sample, oil sample, in test tube and 2 ml of chloroform and shake well and observe that sample completely dissolves in the chloroform. That also confirms, that is in principle we have said that uh, lipids are soluble in organic solvents, and you may confirm there, that sample when completely dissolved with chloroform, that is the lipids are present. Then emulsification test is there. We take 5 drops of sample in test tube plus 4 ml of distilled water plus 4 ml of 5% sodium carbonate and shake well. The lipids are uniformly distributed, therefore solution forms an emulsion. When we shake the test tube with these particulars, the uniformly distributed droplets of the lipids, you may see that confirms the emulsification of the oils, and emulsification is itself indicates that the lipids are present in the sample. So all these 3 tests confirms the lipids are present in the sample. So this is one more practical given for me, that is identification of sugars by ascending paper chromatography. That is when we take sugars are separated with the help of paper chromatography. In principle, we can say that simple and inexpensive analytical technique for the separation of the organic and inorganic substances. In paper chromatography, mobile phase is a liquid containing solvent system, and in solvent we have to put some stationary phase. That stationary phase is the Wattman number 1 filter paper. The mixture to be separated is applied on the spot, as a spot on the end of the chromatography paper. The paper is kept in a glass jar. After sufficient running of the chromatogram, it is dried with the help of dryer and then sprayed with the reagent for identification of the spot. The position of the different components from mixture is determined by the calculating RF values, and RF value is calculated by the formula distance traveled by solute divided by distance traveled by solvent. Then for this purpose, requirement of Hartmann number 1 filter paper is there. Cylinder, beakers, capillary, all that you may know. Mortar pestle for the crushing of the material. Chemicals like ethyl alcohol, butanol, glacial acetic acid. I have made here 3 to 4 systems for the. I have collected 3 to 4 systems for the sugar, but yet not tried. You have to take the initiative, some of our colleagues, and see whether the which is the correct system occurring here. I prefer for the N-butanol, acetic acid, and water. For that purpose, aniline phthalate and p-anisaldate, these are the 2 spray reagent. Standard glucose mannitol are used for this practical as a standard solutions. How to prepare the standard solution for sugar is the solutions were made roughly 1% in concentration, except in case of lactose and maltose, where we need some more amount, that is up to 5%. That such prepared material is loaded on the paper with 3 to 4 microliter per sample, not more than that. Plant material is fresh leaves of the Gossypium, Sesbania, etc. You may choose here other sources of the sugar, like gum or other sources, but I prefer Gossypium, Sesbania, Bryophyllum leaves. take 5 grams of leaves of any selected plant material, then wash them with the water and blot to dry, crush them with the 80% alcohol, filter through the Buckner funnel section, take the filtrate as like the other practicals of the we have to take physiological practical. Evaporating dish is taken and condense it for the 1 to 3 ml, cool it to room temperature, and 2 to 3 ml distilled water is added afterwards. Mix it well and centrifuge again for 1000 rpm. For sugars, the supernatant acts as extract for the sugars. Preparation of chromatography paper. Origin is marked at the distance of 0.5 centimeter to 1.5 centimeter. End is marked at the 10 centimeter on the chromatogram. Loading on chromatography paper using you may use micropipette also, or you may use capillary. With the help of micropipette, you can load appropriate amount of the sugar solution, plant extract or standard sugars. That at the mark the origin at equal distance, you have to place the spots and that to the perpendicular to the paper. The different spots are prepared using separate capillary for each sample. After this chromatography paper is dried and with dryer or in sunshine. Then the solvent system is prepared. I have collected 3 to 4 solvent systems, but I prefer butanol, acetic acid, and water. The laboratory of your choice or your laboratory, which material is suitable, which solvent system is suitable, that you may try. For first solvent system, butanol, acetic acid, water, 4:1:5 proportion is there. You have to take the lower portion of the solvent system. The spray reagent is for this particular type of solvent system is aniline phthalate. The other 3 systems are there for which you can use p-anisaldate as the spray reagent. For benzene, butanol, acetone, water, you may take in 20, 30, 14, 10 by volume. Then other 3 are there. You may visualize on the screen. Then running of the chromatogram, as all of you are knowing that we are trying this practice from last several years to show chlorophylls and all other pigment like organic acids on the paper chromatography. Then this is plant extract is loaded. Plant extract is loaded on the paper, kept it in vertical position in the cylinder that we have we are knowing. Load spots at origin above the level of the solvent system. Precautions may be taken like this. The cylinder is made upright, airtight cylinder is made. Then remove the chromatogram paper when solvent system reaches near the end. Development of this chromatographic paper is with 2 sprays. First spray is prepared to dissolve 1.8 gram aniline and 1.8 gram diphenylamine in 80 ml of acetone, and add 20 ml of the concentrated HCl. Spray with 0.93 gram of aniline and 1.66 gram of phthalic acid. Dissolve in 100 ml of N-butanol saturated with water. So this is first spray region you may use for the you should use for the N-butanol glycolacetic acid and water solvent system, and that is that system is famous as BAW system. And spray reagent second is for the it is 1 ml of freshly prepared 1 ml of p-anisaldate. How to prepare that? 1 ml concentrated sulfuric acid, plus in 20 ml of ethanol, heat at 110 degrees centigrade. Sugar phenyl hydrazone gives green yellow spots in less than 5 minutes. That confirms the sugars. You may count the number of the sugars and you may write in the table or in the according to the RF values of the sugars. I have collected two RF values from two references, Rao and Berry, 1951. Arabinose glucose. You may choose 26 glucose and sucrose. Any fructose you may have in laboratory or glucose. For these two, there is a single value, that is 0.26, and with second spray reagent and all other three solvent systems, we have these RF values. For circular, there are also values. For straight or upright chromatographies, there are also values. D-glucose in this system, 0.39 is the value, and if you choose other like maltose, there is the value is 0.36. So these are the RF values. You may tally your RF values with this plus or minus a 2 difference is acceptable. Next practical is to determine the fatty acid value from the sample. Lipids are the naturally occurring compound that we have seen. Plants like seeds, groundnut, plant seeds like groundnut, coconut, sunflower, castor, which are rich in lipid, may be used. The same I have taken here the principle that then the when we fats or oils are stored in the longer times, the produces rancid smell, or due to hydrolysis of the lipids, it produces rancid smell. Or sometimes microorganisms act on that, and the action of the atmospheric oxygen produces peroxide, and that also gives rancid smell. The amount of fatty acids present in the fats or oils at any given time can be indicated by this fatty acid value. Why we calculate the fatty acid value? The fatty acids are amount of fats which are present in the oil are calculated with the help of fatty acid value. So it is the amount of alkali required to neutralize fatty acids present in 1 gram of fatty acid oil. These are the alkalis like NaOH or KOH. The KOH is used. The amount of alkali required to neutralize fatty acid value present in 1 gram. These may count the fatty acid value of the sample. Then the value indicates quality of the lipid, and hence it is important commercially. The fatty acid value known amount of the oil sample is calculated by using titration and using standard alkali with the example, that is KOH. KOH is taken in 0.1 gram, 0.1 normal, that is dissolve 5.611 gram of KOH in the 100 ml of distilled water, 1% phenolphthalein indicator in ethanol. Neutral solvent is prepared with the help of 25% ether, mix 25 ml of ether, 25 ml of 95% alcohol, and 1 ml of 1% phenolphthalein solution, and neutralize with 0.1 normal alkali. So this gives us a neutral solution. Plant material, oil sample you may use any. Here I have given groundnut oil and coconut and soybean oil. 5 gram of sorry, accurately weighed 5 gram of oil sample is taken in a clean and dry conical flask, and 50 ml of neutral solvent and few drops of phenolphthalein indicator in it, and titrate against the 0.1 normal KOH. Care should be taken that continuously shaking the conical flask is needed, and at the end point, colorless to faint pink you may observe and record the reading. This reading indicates the amount of alkali and required to neutralize the free fatty acids present in the sample. For to calculation, 5.611 grams of are present in the 0.1 normal, 1 ml of the 0.1 normal KOH. You may calculate this with the formula. We'll later on discuss about it. Separate amino acids with the help of TLC, as like the TLC of the paper chromatograph of the whatever sugars. We have to take some precautions with the TLC of the amino acid separation with the TLC. Then chromatography technique. Thin layer chromatography is a simple and sensitive, inexpensive technique. Separation of organic acids and inorganic acids. TLC is the phase. In TLC, mobile phase is liquid containing single solvent mixture of the solvent preferred, and stationary phase is the active fine solid known as absorbent. Absorbent. A thin layer of absorbent is fixed on the plate with the binding agent such as gypsum or CaSO4 or starch. The mixture to be separated is applied as a spot at the near the end of the plate. Then the cylinder plate is kept in the cylinder and allowed to run for the sufficient time. And after running at the end, when it reaches, you may take out the plate of the TLC and dry it in air or spray. Dry it in air or with the help of oven. You may dry. Then spray it with the spray reagent, and spots were visible. The positions of the different spots are determined by calculating the RF values. The value is given in the form. RF value is calculated distance traveled by solute, distance traveled by solvent. In that format, you can calculate the distance. For this purpose, all of you are knowing that the glass plate is needed of appropriate size, glass jar, measuring cylinder, etc. All these are the necessary requirements: mortar pestle, silica gel, N-butanol, glacial acetic acid, neem hydrogen, chloroform, etc. are needed. Fresh leaves of Sesbania bryophyllum you may choose, or some may choose infected leaves of beans. Then the preparation of plant extract is take 5 grams of mosaic or infected leaves of the or Sesbania leaves of the fresh leaves of the Sesbania or bryophyllum leaves. Wash them with the water and blotted to dryness. Crush them in mortar pestle with 80% alcohol. Other procedure is same, that evaporate material, then take cool it to room temperature, then mix it well and centrifuge with the help of centrifuge machine. And supernatant is used as for the extract of the for the amino acid as a source of amino acids. How to prepare silica gel slurry? That we are knowing that a silica gel slurry is made and in the chloroform, poured in carefully and press gently with the glass rod so that the uniform slurry is spreaded, or you may have a one hand the excess slurry is wiped out. Then origin marked as 0.5 centimeter to 1.5 centimeter, preferably 1.5 centimeter, because 0.5 centimeter is not suitable for the TLC. It is okay in the paper chromatography, but in TLC you need to have some more distance, and so that a size of 1.5 centimeter is left at the origin region. The loading of chromatography plate by using micropipette, or you may have with the help of a capillary. Spots are loaded on the chromatography table, chromatography TLC at the origin at equal distance perpendicular to the length of the plate. Then the different spots are prepared using separate capillary, because it is very essential that mixing of the material should not be there, or standards should not be there. The preparation of solvent is the famous solvent system, that is butanol, acetic acid and water is taken in proportion of 80, 20 and 20 respectively. The sufficient amount of, when running is there, sufficient amount of solvent system is prepared and is placed in a vertical or cylinder. Vertical cylinder means that cylinder jar, glass jar. Running chromatograph is like that. The plant extract, when loaded on the TLC plate, that plate is kept in the vertical position in the cylinder containing solvent system. Then load spots on different positions by keeping the distance, and the cylinder is made up airtight. Then remove the plate when it is appropriately reached to the near to the end. Then developing of the chromatograph plate or chromatographic plate. Plate is air dried, and after that the neat hydrogen solution is sprayed. Then wait for dried, and the development of the violet purple bands will be visible. That confirms the presence of the amino acids. Calculate the RF values. And that confirms the following. There are some amino acids in the plant extract. So one more practical is given to me, that is isoelectric point in plant material. The practical was like this. For determination of isoelectric point in from the material, we have to take proteins from the different sources. Proteins are made up of hundreds of individual amino acids that we are knowing. Various kinds of proteins are there. Each may have positive and negative charge depending on the pH of the system. At home, pH value, all the positive charges and all negative charges which are on the proteins, charges on the protein will balance, so that net charge on the protein will be 0. So isoelectric point of the protein is the pH at which the protein is electrolytically immobile. So at this particular pH, there is no charge on the amino acids. So there is a requirement of chemicals like acetate buffer, 3 pH 3, pH 4, pH 5 and 6. Then phosphate buffer of pH 7, 8 and 9, 10. 0.14 molar NaCl solution. For that purpose, you take 4.0 gram or approximately 4 gram of NaCl in 500 ml distilled water, and 0.1% casing solution. So Plant material is used is seeds of Azuleus aconitifolia matki. You may use for that purpose. There is a procedure for extraction of the proteins. The germinating seeds were washed thoroughly, blotted to dryness, and remove seed pods. 1 gram accurately weighed 1 gram of the seeds crushed in the 10 ml of the cold 0.14 molar NaCl solution. The extract is filtered through double-layered muslin cloth and then centrifuged if necessary. Then supernatant is used as a source of protein extract. And assay is prepared by taking a set of 3 test tubes and label as shown in the observation table. That test tube as first test tube as acetate buffer pH 3, second test tube as acetate buffer pH 4, and third test tube as acetate buffer pH 5. Then at pH, then say take 5 ml of acetate buffer in each test tube and 5 ml of phosphate buffer in each test tube. Then add 1 ml of plant protein sample in each and observe the pH value at which the maximum precipitation is observed. Here, an example is given that at 4 pH, at 4 pH we have maximum precipitation of the proteins is observed, and so repeat the experiment for different proteins. You may, if you want to do. The isoelectric point of casing is 4. So casing is used in this practical. The isoelectric point protein from given plant material is 4. So this is all about the isoelectric point of protein, and you may follow this procedure for the calculating isoelectric point of protein. Last practical in my fourth practical is visit to nursery, aesthetic value, aesthetic gardens, exhibition, food industry. You may visit any of the nursery industry, and then students have to submit the report in a separate sheet. One more practical is there, practical number 3 from 3, that is use of dialysis of smaller molecules from larger molecules. This is the total new practical, and that practical is now I have collected only information about the this practical. It is totally new to me also, because that practical has not done by me. The information is collected only. The principle dialysis of smaller molecules, use of dialysis for separation of the smaller molecules and larger molecules in a solution. The dialysis process by which small molecules in a solution are separated from the large molecules. The principle behind this is discovered by the Scottish chemist, that is Thomas Graham, in about 1861, and Graham found that the rate at which some substances such as inorganic salts pass through a semi-permeable membrane is up to 50 times as great as the rate at which the other substances, such as proteins and other chemicals, organic molecules. We know that such rate differences depends on the fact that the openings in the semi-permeable membrane are very nearly to size of the atom, ions, and small molecules. That makes it possible to pass through the semi-permeable membrane. So I have collected much information that the 1% NaCl is needed, that I have taken in 1% solution. Corn syrup solution, water, vinegar, glasswares like dialysis tube you may use, that is beaker, stirring material you should use, and some rubbers, string material like you may use rubbers. In place of string material, you may use rubbers. Plant material like sugarcane juice, corn flour, you may use, or you may use the sugar solution directly by sucrose, adding sucrose. Other material is egg. Procedure is like that. Prepare sugar and salt solution using suitable material. Any of the material you may use, and sugar and salt solution is prepared. Preparation of dialysis tube using egg. Prepare dialysis tube by dipping the egg in vinegar. After removing the yolk from the egg, so as to remove eggshell and other outer covering of the egg. Use use it as a dialysis tube, or use dialysis tube from the market. You may remove the shell of the egg, and you may have egg membrane. The thin membrane is visible. Use that membrane as dialysis tube. Tie the tube after filling the above sugar and salt solution in it. Keep the dialysis tubing in the 3/4 filled beaker as shown in the figure. In this figure, I have shown you that this is the level of water, or you may call it solvent. This is the tube which is prepared from the egg membrane. Then, as time passes, the material that is which is kept inside the tube comes out of the membrane, dialysis membrane, and comes in the water. So for that purpose, there is one CT, but I have not still given here. Movement of the smaller molecules takes place from tube into water. After 30 minutes, observe the change in the solution present in the beaker. Then I have taken rise in for that purpose, a CT is taken for Cl with the AgNO3. Result is that from solvents, ion and buffer can diffuse easily across the semi-permeable membrane, but larger molecules are unable to pass through the pores of the semi-permeable membrane. Thank you for patient hearing. Thank you one and all. Thank you Dahiwadi College, Dahiwadi, for inviting me as a resource person and sharing my views with you. And thank you all team of the Dahiwadi College, Dahiwadi. Thank you one and all.
Okay, thank you so much, sir, sharing your practical knowledge with us. We kept your terrific talk on this practical in our mind. Thank you so much, sir. Now, chairperson for this second session is respected Professor S.K. sir, Head Department of Botany, PDVP College, Tasgaon. Also, visiting faculty member of Department of Botany, Shivaji University, Kolhapur. 2 PhD candidates are awarded under his guidance, and 4 candidates are working. He published 40 research papers in national and international conferences. I would like to welcome, on behalf of Department of Botany, Dahiwadi College, Dahiwadi, I would request to
thank you, madam. Thank you,
Professor Khade sir, to give his opinion and suggestion regarding this. Yes, sir,
thank you,
please.
madam. I am audible. I am audible,
yes,
madam.
Sir. Yes, sir. Yes, sir.
Okay. Thank you. Good afternoon, all of you. Myself, Professor Dr. Khade S.K. from PDVP Mahavidyalaya, Tasgaon, Sangli. I am very much thankful to principal and the team of Dahiwadi College, Dahiwadi, for appointing as a chairperson for a session of physiologist Dr. B.A. Sonar from Department of Botany, Balwant College, Vita. I am here to concluding session, concluding lecture by Dr. Sonar. Sonar gives very good information and knowledgeable lecture on the B.Sc.III revised practicals, paper number 3 and 4. I am very thankful to the different lecturers and different colleges students from different colleges affiliated to Shivaji University, Kolhapur. So again, I am just overlook the total program. Early in the morning, the session opened by Professor Dr. Jadhav Madam from Head of Department, Shivaji University, Kolhapur. After this session, the senior professor Kamble sir gives the best wishes for this program. Then give the keynote address by Dr. M.B. Wagmare sir. He is the chairman of Botany, the Shivaji University, Kolhapur. So again, I am just overlooking the today's lecture from the Dr. Sagar Deshmukh, New College, Kolhapur. Second lecture by Jadhav sir, Varna College, Varna. Then Moholkar sir, Aund College, Aund. Then last one, the Dr. B.A. Sonar from Balwant College, Vita. So Sonar sir gives the very informative lecture, informative knowledge about the different types of practicals. They are included in the biochemistry, the practicals such as lipids, proteins, concerning to that proteins, amino acids, then cellulose, then the different lipids, then paper chromatography, fatty acid, then thin layer chromatography, and last one is the isoelectric point. So, and again, the last practical, that is the nursery visit. Just with your kind permission, I am talking in some views in Marathi. सकाळपासूनच्या डॉक्टर जगताप आणि विवी कांबळे सरांनी सकाळपासून घेतलेले एफर्ट्स अतिशय चांगल्या पद्धतीनं शेअर केले, चांगल्या पद्धतीनं एक्सप्लेन केलं. आणि या सगळ्यामध्ये सगळ्यात चांगली गोष्ट काय असेल तर आजच्या ह्या प्रॅक्टिकलमध्ये, प्रॅक्टिकल एक्सप्लेनेशनमध्ये किंवा प्रॅक्टिकल वर्कशॉपमध्ये जे रिसोर्स पर्सन चूज केले ते अतिशय चांगल्या पद्धतीनं आणि चांगले होतकरू नवीन मधले सागर देशमुख असतील किंवा मोहोळकर असतील किंवा जाधव सर आणि बाबू सोनार तर माझे गुरुबंधू आहेत. आणि या सगळ्यांचं अतिशय चांगल्या पद्धतीनं त्यांनी सरांनी सिलेक्शन केलं. आणि खरं तर मी या सरांना, अनिल जगताप सरांना 2-3 वेळा बोललो की सर नवीन लोकच घ्या या चेअर सेशन मध्ये. तर मला त्यावेळी त्याने आग्रह केला की सर राहू दे, की तुमचा सुद्धा काही या संदर्भातला व्ह्यूज काय असतील, काही गोष्टी तुम्हाला शेअर करता येतील म्हणताना मी हे स्वीकारलं. आणि परत एकदा आय एम व्हेरी थँकफुल टू दी टीम ऑफ दहिवडी कॉलेज, दहिवडी, आणि स्पेशली द प्रिन्सिपल सर and Anil Jagtap sir and Vivek Kamble sir and the other colleagues of Department of Botany. Once again, I am very thankful to the Devadi College. Okay, bye.
Okay, thank you so much, sir, for your diamond talk. Now the session is open for discussion. I think there is no question and queries in chat box.
Okay, madam. Okay, thank you.
Okay, so once again, I would like to thank a resource person of this session, Dr. Moholkar sir and Dr. Sonar sir, and chairpersons Dr. Rekha ma'am and Professor respected Khade sir on behalf of Department of Botany and our college. Now, I would like to request Dr. V.V. Kamble sir for his valedictory speech.
Nare ma'am. Hello. Nare madam.
येस सर अनम्यूट केलेलं आहे त्यांना.
अच्छा चालू करा.
Hello.
आवाज येतोय सर.
बोला की.
Okay. Friends, now we are here in the last session, that is valedictory session. Today, on the occasion of online workshop on revised syllabus of B.Sc.III, practical 3rd and 4th, here we are participating in this session, and today we have Principal Dr. S.D. Helkar sir for this session as a chief guest, and Principal Dr. B.S. Balwan sir is the president for this session. I would like to introduce Principal Dr. Helkar sir. Principal Dr. S.D. Helkar sir is presently principal of Professor Dr. N.D. Patil College, Malkapur. He has completed his MSc and PhD from Pune University, and his area of specialization is Ascomycetes taxonomy. He has a teaching experience of 18 years with research experience of 10 years and administrative experience as a principal in Rare Shikshan Sanstha's various colleges for last 13 years. There are more than 20 research papers in his credit in reputed international and national journals. He has attended more than 120 conferences, workshops, symposia, and seminars. In addition to this, he is also a member of scientific societies. With this brief introduction, may I request Honorable Principal Dr. S.D. Helkar sir to guide us on this auspicious occasion. Thank you. Thank you very much. Over to Principal Dr. S.D. Helkar sir.
Hello.
आहे नंबर आहे नंबर आहे कोण लिखत हे किसकी है?
सुहास हा साहेब बोला. हॅलो हॅलो त्यांचा आवाज येईल का प्रेसिडेन्शियल रिमार्क घ्यायचं म्हणतो.
जगताप सर,
हा बोला.
काय सरांना अडचण आहे का काही?
आपला आवाज त्यांचा आवाज येईना झाला आपल्याला जॉईन झाले.
हा मग काय ती माईक वगैरे त्यांचा आहे का तिकडं म्यूट अँड म्यूट काय असं?
त्यांना सांगितलंय मी लिव्ह व्हा आणि पुन्हा जॉईन व्हा.
हा मग तसं थोडंसं झालं पाहिजे ना, तरच ते.
हो हो सांगितलंय त्यांना मी आता. 1-2 मिनिटं वेट करूया आपण साहेब.
ठीक आहे चालू.
इथं अनम्यूट आहे का? किती आलं का?
साहेब बोला. हॅलो बोला साहेब. 2 चालू करू नका साहेब. दुसरं कोणी चालू केलं असेल तर त्यांना थोडंसं. हा साहेब बोला.
म्हणजे बंद करा.
हा साहेब बोला. हॅलो
चालू कर.
हा साहेब आवाज येतोय बोला. हॅलो
व्हिडिओ स्टॉप करा.
हॅलो
हा बोला साहेब.
हॅलो
हा बोला. हॅलो हॅलो हॅलो
हॅलो
आवाज येतोय साहेब.
हा सर.
येस येस आवाज येतोय.
जगताप सर आवाज येतोय का?
हा हा येतोय.
हॅलो
येतोय येतोय आवाज येतोय.
हा येतोय.
ह्याच्यामध्ये हा जगताप सर.
हा येतोय आवाज.
हॅलो
येतोय आवाज सर. आवाज येतोय
आवाज येतोय का?
तुमचा. हा येतोय साहेब बोला.
आवाज मोठा.
स्वतःला अनम्यूट करून साहेबांना सांग आवाज येतोय म्हणून ऑडिबल म्हणून. तुझ्याकडे घे हँड ओव्हर सेशन. माझं बॅटरी गेली. हॅलो
हॅलो सर.
हा येतोय येतोय.
जगताप सर.
आवाज येतोय साहेब तुमचा बोला.
अच्छा ओके ओके ओके. नमस्कार. गुड आफ्टरनून एव्हरीबडी. The love for all living creatures is the most noble attribute of man. I am Principal Dr. Sunil Helkar from Ray Shikshan Sansthan, Professor Dr. N.D. Patil, Mahavidyalay, Malkapur, Kerala, Taluka Shalwadi, District Kolhapur.
बॅटरी गेली लहान त्यामुळे मला कळालं.
Everything is already discussed about practicals in these sessions. Now, without discussing about ongoing sessions of practicals, I am to focus on need of awareness about conservation of biodiversity in Western Ghats. Friends, being a teacher of botany, I have great opportunity to work in Professor Dr. N.D. Patil Mahavidyalay, Malkapur as a principal, teacher, and researcher of botany. Our college is situated on the lap of Western Ghats. Western Ghat is a biodiversity hotspot. As a nature observer, from last 3 years, the plant cover in this area is depleting day by day because of conversion of forest land into agriculture land, cutting of forest plants for timber, and human interference in natural ecosystem. Forest fire in summer season, bauxite open mining at Girgaon, Dopeshwar, and Udgiri areas of this area. Increase in number of quarries. And the last, increase in number of tourists in historical and religious places that will create burden and disturbance on wildlife in this area. The wild animals are coming into the agricultural fields in search of food and forest fodder. That is creating conflict between human and wild animals. It is a day-to-day problem in this area now. It is our responsibility to create awareness about importance of biodiversity, plant cover, and wild animals. I thank organizers of this workshop for giving me an opportunity as a chief guest for this valedictory function. Thank you one and all. Jai Hind, Jai Karmavir. पुढचा काय रिस्पॉन्स नाही. हॅलो
हॅलो.
हा जगताप सर, ओव्हर.
Okay, thank you, sir. Then I will request to Dr. B.S. Balwant sir, Principal of Botany College, Devat, to give a presidential remark. Balwant sir.
ओके सर, ओके. शिवाजी विद्यापीठ कोल्हापूर आणि देवडी कॉलेज देवडी यांच्या संयुक्त विद्यमाने वनस्पतीशास्त्र विभागाच्या वतीनं जी कार्यशाळा आयोजित केलेली आहे, या कार्यशाळेच्या समारंभाचे प्रमुख पाहुणे आमचे ज्येष्ठ सहकारी प्रिन्सिपल डॉक्टर त्या कार्यशाळेमध्ये ज्यांनी विषय तज्ञ म्हणून मार्गदर्शन केलं ते माननीय डॉक्टर देशमुख सर, यादव सर, डॉक्टर मोहोळकर सर, डॉक्टर सोनार सर, आणि या कार्यशाळेसाठी ज्यांनी आम्हाला आपला मूल्यवान वेळ दिला ते या प्रत्येक सत्राचे अध्यक्ष सन्माननीय डॉक्टर भालेराव सर, माननीय श्यामसन गायकवाड सर, माननीय डॉक्टर रेखा देवकर मॅडम, डॉक्टर खाडे सर, आणि या कार्यशाळेचे संयोजक प्राध्यापक डॉक्टर जगताप सर, विभाग प्रमुख प्राध्यापक कांबळे सर आणि त्यांचे त्याबद्दल मी महाविद्यालयाच्या वतीनं शिवाजी विद्यापीठ कोल्हापूर आणि BOS मंडळाचे चेअरमन आणि अध्यक्ष सदस्य यांना धन्यवाद देतो. आणि या कार्यशाळेमध्ये सकाळपासून शिवाजी विद्यापीठाच्या कार्यक्षेत्रातील सर्व वनस्पतीशास्त्र विभागाचे प्राध्यापक बंधू भगिनी ऑनलाईन जॉईन झालेले आहेत. मला निश्चितपणे या कार्यशाळेच्या निमित्तानं माझ्या मनामध्ये आशावाद आहे की या कार्यशाळेच्या निमित्तानं शिवाजी विद्यापीठातील सर्व सहकारी प्राध्यापक बंधू भगिनींना चांगल्या प्रकारची माहिती मिळालेली आहे आणि याचा निश्चितपणे जो उपयोग आहे ते विद्यार्थ्यांना होईल. आज आमच्या महाविद्यालयातील या वनस्पतीशास्त्र विभागाला सर्वांनी सहकार्य केले त्याबद्दल मी महाविद्यालयाच्या वतीनं आपणा सर्वांना धन्यवाद देतो. आणि आमच्या महाविद्यालयातील या विभागानं कार्यशाळेचं अतिशय सुंदर नियोजन केलेलं आहे. त्याबद्दल विभागाचे अध्यक्ष, विभाग प्रमुख प्राध्यापक डॉक्टर कांबळे सर, त्यांचे सर्व सहकारी यांना धन्यवाद देतो. आणि या समारोप समारंभासाठी रयत शिक्षण संस्थेच्या डॉक्टर ND पाटील महाविद्यालय मलकापूर येथील आमचे ज्येष्ठ सहकारी प्रिन्सिपल डॉक्टर केळकर सरांनी आपला मूल्यवान वेळ दिला त्याबद्दल त्यांना धन्यवाद देतो. आणि खूप वेळ चाललेली या कार्यशाळेसाठी मला अध्यक्ष म्हणून या विभागानं आपलं मनोगत व्यक्त करण्याची संधी दिली त्याबद्दल वनस्पतीशास्त्र विभागाला धन्यवाद देऊन या ठिकाणी माझा अध्यक्षीय मनोगत थांबवतो. धन्यवाद सर.
Thank you, sir. So this is now time to propose a vote of thanks. So firstly, I propose a sincere thanks to University Authority to giving this opportunity to organize a online workshop on a revised syllabus of B.Sc.III practical number 3rd and 4th. So once again, I thanks to this authority. Then I also propose a vote of thanks to Professor Dr. V.D. Jadhav ma'am, Head Department of Botany, Shivaji University, Kolhapur, for his valuable guidance in an inaugural speech. Again, I thankful to my research colleagues, Dr. M.B. Magmare, BOA Chairman in Botany, Shivaji University, Kolhapur, as well as I propose a vote of thanks to resource person, Dr. S.A. Deshmukh sir, Dr. S.Y. Jadhav sir, as well as my teacher, Dr. S.M. Molkar sir, and my friend, Dr. B.A. Sonar, for this acting as a resource person, as well as I also propose a vote of thanks towards the chairperson, Dr. E.B. Bhalerao sir, in charge principal, Kishanvir Mahavidyalay Y, Mr. Samsung Gaikwad sir, Head Department of Botany, YCIS Satara, Dr. Rekha Devkar ma'am, POS member and Head Department of Botany, Deshbhakt A.B. Naik Arts and Science College, Chikli, as well as the 4th session person, Professor Dr. S.K. Khade, Head Department of Botany, PDVP College, Tasgaon, and the chief guest of the valedictory function, Principal Dr. S.D. Helkar sir, Professor N.D. Patil, Mahavidyalay, Malkapur, as well as the our beloved principal, Dr. B.S. Balwan sir, for granting the permission to organize such a type of the workshop, and my all the colleagues in the Department of Botany. And I also propose a thanks towards all the participants, heads, and BOS members of Botany in Shivaji University, Kolhapur. Last but not least, I propose a vote of thanks to one and all. Thank you very much. Thank you. Okay. Okay, thank you. सुहास सर.
Yes sir, लेफ्ट करतो, एंड करतो मीटिंग.
ते चॅट बॉक्स मध्ये लिंक टाकलेली मेसेज फक्त जाऊन.
हा पाठवले.
ओके थँक्यू सर. आता लिव्ह करा सर. ओके
एंड करतो सर मीटिंग.